So Jack May's our first speaker, and he comes to us from Cal State, Long Beach, and he works with Dr. Shark, the Shark Lab. The whole school got renamed.
Yeah, apparently we're the Sharks.
The Sharks,
yeah.
They went for, yeah. So.
Go Beach.
Go Beach, go Sharks. So good. Okay, well, I'll let you take it away. Hopefully.
All right, good afternoon, everyone. Today I'll be talking to you about my research with leopard sharks out at Caliana. That way. Okay. All right, so yeah, first I'd just like to start by talking about aggregation behavior with animals kind of in general. So from an evolutionary standpoint, basically the benefits of them coming together into these groups need to outweigh any negative costs associated with that behavior, right? So some of the benefits can be for social reasons, get together, kind of communicate a little bit more easily, can be for reproductive, so get a lot of individuals together, a little bit easier to find a mate, or to exploit a resource. This resource can be something like a desirable prey item, or maybe even just a desired type of habitat. And then a downside can be for increased disease potential, so like viruses, parasites, anything like that. If you're living in close proximity, it can spread a little bit faster, as well as competition. So if there's a limited amount of this resource and more individuals start coming into the area, it might not be enough to go around, it can kind of start spark competition, things like that. So, turns out leopard sharks, here in the bottom right, they happen to perform this aggregation behavior. So why is it that they might be doing this? So, some previous work has shown that leopard sharks during like the warmest summer months, it's not uncommon for them to come together in big numbers in really shallow water near shore. So if you look at this light column right here, so basically these are during the daytime, and you can see that temperature would rise during the day, and then at night it would fall. So nothing mind-blowing, if you look at the average number of sharks, which is the black diamonds, and then the circles, which is average temperature, as average temperature increased, so too did the average number of sharks. And then it would decrease later on in the evening. So sharks are looking for warm water. Why might that be? So turns out it's mostly mature females, most of which they've checked turned out to be pregnant. And they're thought to use that warm water. They're ectotherms, so if they can increase their body temperature, they can kind of speed up metabolism and increase that gestation rate and shorten how long they're pregnant, brood those little pups. So then at night, they move into cooler, deeper water, thought to be foraging. They might even move to like other aggregation sites the next day. So some of the ones that were tagged at Catalina Island made their way all the way to like La Jolla, so they don't necessarily come to the same site over and over and over again. So some of the big aims for this project are basically to quantify the location of the sharks in relation to seafloor temperature. The way I'm going to do that is basically fly surveys using a drone, and then also couple that with seafloor temperature that I'll collect using little temperature loggers. And then secondly, to look more at the social aspect of the aggregations, doing that using high resolution footage from the drone flights to basically quantify some of the characteristics of the aggregation, like average size of individuals present, distance between individuals, things like that. Okay. So for this summer, basically I spent almost all my time at Catalina Harbor. There were some sharks recently spotted in Big Fisherman's Cove, but the most I've ever really heard of is about like 15, most of the time onesies or twosies. So kind of left that alone. Emerald Bay is where I spent most of my time last summer, and they have had some sightings this year, but every time I've gone to check it out, I haven't seen a whole lot. So pretty much been sticking to Catalina Harbor where I can pretty much safely see like 100 sharks easily like every single day. So conditions are kind of touch and go, but lots of sharks, so it's really nice. So for my flights, I use a Phantom 4 Pro. It's from DJI, pretty good stuff. I ended up flying over 55 hours this summer, over 160 flights, and kind of still counting. I'm still out there taking flights when I can. And then to collect seafloor temperature using the Hobo temperature loggers, basically spaced out a little semi-grid pattern at the sites, but I'll talk a little bit more about that. So first, for the surveys, that's Cat Harbor right there. Basically, that northernmost end right here, and then over here by the yacht club is right where I've seen the most sharks. You can kind of sort of see some of them right there. But anyways, so the surveys are typically flown in 3 segments. So the first one's at like my relatively like highest altitude, around 25 meters, basically just trying to identify where the sharks are. And then once I've identified where sharks are, I basically fly over each discrete aggregation for about 5 minutes. And then I'll go down to about 10 meters altitude where I can get my highest resolution video. And again, that's going to be good for looking more at like getting size estimates and looking at more of like the specific behaviors. Then I'll take those drone images and mix that with temperature maps that I'll have made. And the way I basically do that is I lay out these little yellow concrete discs. You can see kind of spaced throughout here. Each one of those has a temperature logger on it, and they log together every 10 minutes. And then once I have that temperature data downloaded, I can then basically like use a variety of interpolation techniques to create a prediction of the seafloor temperature. So here you go. So this was last summer in Emerald Bay, and you can see here in the shallow water, it's going to heat up like in the so this is probably around like 2 o'clock in the afternoon. So you can see this is heated up in the shallows and still stay cool in like the deeper water. So this is basically what's causing potentially these sharks to aggregate in shallow water. So that's like a limited area of warm water that's available to them. And then here's a little animation to just kind of show that over time. You see again the warm water here, like in the most shallow areas, and then in the evening and early mornings, it all kind of becomes uniform. So it kind of helps see why they kind of aggregate during the day and then disperse in the evening time. All right, and then here's just a quick little video of an example. Again, it's Emerald Bay. Got one aggregation here, and then arguably another discrete aggregation right here. You can see all the concrete discs in the water, and they serve a role as putting the temperature loggers on the bottom, but then also as a known sized object. So I can use that known sized object to basically do size estimates. So using things like ImageJ or Adobe Photoshop, basically calibrate the screen to however many pixels, to however many centimeters, and get a pretty good estimate of how actually long those sharks are. All right, so some of the preliminary data that I just have from some of the images I've gone through from this summer's flights. So here's Cat Harbor by that yacht club, and you can see all the sharks kind of right here along the beach. So once I've like kind of geo-referenced that image and put it on the like in the world, like where it should be, I can then basically mark the location of each one of those sharks and then overlay that on the temperature map. So here you can see all the sharks kind of plotted in there, and then apply that to like a frequency distribution where this is in quarter-degree increments. Basically place those sharks and what temperature they were occupying. So this was just for like one sample, but I do this like many times for each aggregation of about 5 minutes. So here's an example from Emerald Bay last year. So again, you can kind of see that the sharks ended up roughly in that 23 to 24 degree range again. And I'll run this is for about a 5-minute period, sample 25 times. You can kind of just get a feel for where those sharks were over that time period. So although you kind of see some individuals come and go, most of them hang out in that one particular area. So eventually I'll look at just what those temperatures were throughout, as well as just to get an idea of like area usage as well. Okay. So another thing we're working on is the automated tracking. So basically having the computers do a lot of work for us, working with some computer scientists. And basically what the goal is to have it be able to identify the sharks and then frame by frame continuously track them throughout the time that I was hovering over them. And then what we want to do is look at things like the size of individuals, which ends up kind of being like a blob of pixels more than anything. But you can look at like their velocity, interpersonal distances, how close they were willing to like be to each other, especially as like density increases, speed, things like that. Trying to see if there's any kind of like size-based dominance hierarchy to compete for warm water. All right, so sharks and drones, pretty cool, but like why should we care, right? So taking the next step and understanding their aggregation behavior, is there like a lot of social interaction going on, or is this just animals trying to get that warm water and that's it, get out of my way? So just taking the next steps and like moving drone methods into ecology, as well as leopard shark is a high trophical species. It's also been shown to be vulnerable, especially like in the early 1990s, late 1980s, nearshore gillnet fishery. Plus you have all these pregnant females hanging out in really shallow water together, very densely near humans, right? So obviously can get impacted pretty quick, pretty vulnerable. So the more we know about them and like what specific temperatures cause that aggregation behavior, especially with like climate change and ocean temperatures, warming and changing in general, get a better idea of where they might be going in the future and make sure the proper management is in place. And with that, I'd just like to thank volunteers, all the fellows for helping out, SCAS from ISCT, and Wrigley, nonetheless, USC, for having me out there. Couldn't have done it without everybody's help, so thank you very much. If you have any questions, let me know.
With the temperature loggers, do you anchor them down or do they move it all with the sand?
So it's, so they're on concrete discs, and I have seen some movement. And so basically what I'm trying to do is with like some of the higher, basically like all those flights had like a high altitude at some point. And so basically I can layer those maps on each other, like those images. And so over time I can kind of update their positions as they did ever so slowly move, Every once in a while a fisherman will like leave one on the beach and I'll kind of put it back, but I'll like just leave that out to that point in time.
So my research this summer was to help bridge the gap between laboratory and natural field observations by studying the physiological state of white sea urchin larvae under in situ natural ocean environments. And so most marine animals have complex early life history stages where they're larvae as free-swimming plankton in the ocean, and they're very vulnerable at this stage, and they have very high mortality, so a small fraction of them will get through metamorphosis and survive into adulthood. And there's a lack of research examining biology under these in situ ocean environments, so there's a lot unknown about the cause of that mortality, whether it may be due to predation, transversal away from a suitable habitat, and genetic inviability, or starvation could all be factors for their high mortality. And so for my research, I focused on food availability, and so looking to be able to answer the question if starvation is a factor in their poor survivorship. And alongside that, also looking at the relevance of lab-based physiological mechanisms and if those are able to be extrapolated into their more natural habitat. And so to do this, I use larvae of the white sea urchin, Lidochinus pictus, which you can see in its larval form and also in its belt form. And these are native. They're found often along Catalina and in Fisherman's Cove where this experiment was taking place. And so here are some videos of the in situ system that was developed. And so this took months of interactions with the engineering and machine shop at USC in order to build, and also a lot of valuable input from the waterfront staff at Wrigley Marine Science Center. And so the culture vessel is 20 liters in size, and so the larvae fit in there, and they have a mesh lid on the top and bottom to keep the larvae enclosed. And then you can see the motorized mixing, which ensures a constant flux of seawater, so we can be sure that the water conditions in the surrounding environment are the same as the water conditions inside the vessel. And so the Wrigley Marine Science Center is an optimal spot to do this research for a number of reasons. One, the water there is very clean, so it supports larval growth and development. Also, this particular location is directly next to an ocean monitoring system, which gives us data of temperature, salinity, chlorophyll, pH, etc. And so those are all going to be valuable variables to look at when we're interpreting the data. And then also, the proximity to the lab makes it feasible to do these types of studies because in a matter of minutes, I can take the larvae out of their ocean environment and have them up in the lab to start their physiological analyses. And so alongside looking at larvae in situ, I maintain constant food amounts in laboratory conditions. So 4 treatments at 50, 30, 10, and 5 algal cells per microliter, and then also an unfed larval control. And so larvae, so the experiment started on the 3rd day when larvae reached their feeding stage, and then carried out through the 5th day when they were taken out and analyzed. And so comparing the ocean and laboratory conditions, first looking at temperature, they were pretty similar between the two. So the ocean is, as expected, more variable in temperature and about a degree warmer. And then nextly, looking at chlorophyll. So the chlorophyll can be used as an indicator of the amount of food present in the environment. So if we were to directly compare the amount of Rhodomonas, or the laboratory algae used, at the lowest feeding treatment of 5 cells per microliter, that would convert to about 7.5 micrograms per liter of chlorophyll. So even the lowest feeding treatment at 5 cells per microliter is much higher than the chlorophyll seen in the ocean. And in addition to that, the chlorophyll in the ocean, we don't know what species, we don't know the composition of phytoplankton. So it could be that the chlorophyll is coming from phytoplankton that are the wrong size or digestibility for the larvae. So both those things would suggest that the amount of food in the ocean is much lower. However, in the ocean, they're getting at this diverse mix of species. So it could be that by having this mix of phytoplankton species compared to a single algal species that we give them in the lab could provide them more vitamins and nutrients and could be a more nutritious diet, even though it's a less quantity. So in terms of measurement, looked at growth, biochemical content, respiration rate, and protein synthesis rates. And the reason that these measurements were chosen is because it allows us to make determinations on their metabolic dynamics and their energetic efficiencies. So respiration rate can be converted into ATP units, which can tell us the amount of total energy that an organism has. And so protein synthesis is the most energy demanding process of the larvae, and they allocate over half of their total energy towards that process. So by knowing how much energy they have and the percentage of that that's going towards protein synthesis, and then on top of that, knowing how much protein, so biochemical content of protein that they deposit as mass compared to how much protein they're synthesizing, rather than getting turned over or degraded, can tell us how efficient they are. And so ultimately, these energetic efficiencies can be used to help predict how organisms will do under different conditions of environmental stress, and ultimately their potential for adaptation to environmental change. So firstly, looking at their respiration rates, so each of these bars is from a different cohort of larvae. And so the unfed larvae have the lowest respiration rate. The in situ have higher respiration rate than the unfed. And then all of the laboratory fed larvae have the highest respiration rates. So the in situ larvae are intermediary between the unfed and fed conditions. And now looking at the protein content of in situ larvae, these also follow a similar trend. So the in situ larvae have a higher protein content than the unfed, but lower than any of the feeding conditions. And so also you may notice that it's whether larvae are fed a 10-fold difference in algae, so between 5 or 50 algal cells per microliter, they're still accreting the same amount of protein. And so the reason that that can be possible is due to a difference in their protein synthesis and turnover rates. So for instance, if a larvae can gain the same amount of protein with less protein synthesis, it would be more efficient. And so it's possible that larvae are able to compensate for lower food amounts by changing their biosynthetic rates. And so that leads to the current analyses that are still ongoing right now in the lab. I'm analyzing their current protein synthesis rates to look at their energetic efficiencies and their growth in the ocean to help determine whether in fact larvae are food limited or whether they may be more efficient in the ocean than in the laboratory environment. And so these experiments achieved a proof of concept for the in situ culturing system. So pending a few more alterations that we need to make to the in situ, then we'll be able to build replicate in situ systems so we can have 2 cultures going at the same time for proper replication for experiments. And then overall, I continue looking at the importance of energy or food availability and regulating responses to environmental change, and ultimately being able to predict resilience of marine life under future ocean conditions. So with that, I'd like to acknowledge all of the Wrigley Institute staff and Manhan lab members, and also Matt Reagan and Carl Oberg, who supplied the ocean data. Thank you. So the machine shop.
Oh, okay.
So yeah, Don Wiggins, and yeah. So.
Anybody have a question?
So now I'm going to switch gear a little bit to talk about these tiny little cells, these single-celled organisms in marine sediments. Even though they don't have bones or muscles or a brain, that they can still propel themselves to survival. All living organisms owe our existence to microbial organisms. And these are the essential environmental engineers who make our planet livable. And our ocean is filled with these single-celled organisms. In average, 10 to the 5th to 10 to the 6th microbes reside in every cubic centimeter of seawater. And that number increases by 3 orders of magnitude in surface sediments. The microbial abundance within the sediments generally decreases with depth, but even hundreds of meters below the seafloor, microbial lives still reside. And if we zoom into a cube of sediments, we can see clearly that there is a frame of solid particles and the pore space in between. Depending on sediment types and sediment depth, the porosity, which is defined as the volume, the ratio of volume of pore space in proportion to the bulk volume of the sediment, varies from 0.3 to 0.9. And the pore water that fills the pore space can take up space as much as about 6% of our global ocean. Within the sediments, the majority of microbes live attached to sediment particles. Therefore, the cells that are free-living in the pore water have never been the focus of microbial studies in marine sediments. But my previous studies have found that about 10 to the 7th to 10 to the 8th microbial cells, the concentration of free-living cells in the pore water was about 10 to the 7th to 10 to the 8th cells. And that is 1 to 2 orders of magnitude higher than what's found in the seawater. So even though that they are only a minor fraction of the total benthic communities, they still constitute a huge number. And not only they are abundant, they are metabolically active, even more so than the particle-attached communities. So this project for this summer is to determine what fractions of these free-living cells are motile and whether these cells are chemotactic. So the study site of my choice is Catalina Harbor. It's an intertidal mudflat. And to conduct this experiment in a controlled manner, but also as close as possible to in situ conditions, I used a flow box depicted on the right. So the bottom of the flow box is filled with sediment, and then I allowed seawater to flow into the flow box on the right and over the sediment and out from the left. And also the inlet is connected to the seawater system at Wrigley. So I always have circulating seawater throughout the system to mimic tidal flushing in natural environments. And for the motility assay, I used a method used by Tom Fenchel in the 2008 study. So in summary, 26 gauge needles were filled with anoxic seawater and connected to 1 milliliter syringes. And these needles are inserted into the sediments through the sampling ports on the side of the flow box. And in theory, the motile cells within the sediment is going to swim into the needle over time, and until an equilibrium is reached in and outside of the needle. And to record this procession, for every set of experiment, 6 needles were inserted at the same time, and one by one, these needles were pulled out at different time points. And eventually, the cell abundances within the needles were determined using fluorescence microscopy to determine the abundance of motile cells. Oh, and also, as you can see from this picture, these experiments were repeated for different depths, so to look at motile cells at different depths. And this figure shows the cell abundancy of this concentration of motile cells within the needle over time, and is also it's color coded for depth. So initially, the needle is free of bacteria. So in principle, the influx of motile cells into the needle within the first 15 minutes or so is a measure of concentration of swimming cells surrounding the needle tip at the time of insertion. And from 15 minutes to 1 hour and after, there was an increase of motile cell concentration. So these are the cells that were not motile at the time of needle insertion, but became motile during the experiment. And these are the ones what I call periodically motile cells. So for the rest of the talk, I will be using the term permanently motile cells and the periodically motile cells defined as such. Then in a separate experiment, I determined the total cell abundance within the pore water. Then I divided the concentration of motile cells by the total cell number in the pore water to get the percentage of motile cells at different depths, both for the permanent and the periodically motile cells. So what we can see here is that from 50 to 90% of the cells in the pore water were motile. But the highest percentage of motile cells was not found at the very surface, but at 8 to 10 centimeter. And even though the percentage of motile cells varies with depth, the percentage of the periodically motile cells were pretty consistent throughout depth. So now we know that a large fraction of the pool water communities are motile. My next goal is to determine whether these cells are chemotactic, meaning that whether they can move towards substrates which are beneficial for their survival, such as electron acceptors. Benthic microbes, they can use different electron acceptors for cellular respiration other than oxygen. And in the upper oxidized layers, oxygen, so these are the strong oxidants. So oxygen, nitrate, metal oxides, they can fuel microbial metabolism. Whereas in the reduced layer, we have sulfate and carbon dioxide acting as the primary electron acceptors. And here for this chemotaxis assay, I used nitrate as a chemoattractant to test for the chemotactic ability of the motile cells in both the oxidized and reduced layer. So this setup is very similar to the motility assay, but except for using the anoxic seawater to fill the needle, I used seawater amended with nitrate at different concentrations. So here we're seeing, again, percentage of motile cells for the permanent in blue and the periodic motile cells in orange at a control 50 and 100 micromolar nitrate addition for both the oxidized and reduced layer. So there was an increase from control to 50 micromolar nitrate addition of motile cell percentage, so which tells us that motile cells were indeed chemotactic towards nitrate at 50 micromolar concentration. And the addition of nitrate also increased the fraction of periodically motile cells. However, at 100 micromolar nitrate addition, there was a decrease of motile cell percentage, suggesting that high concentration of nitrate might be a chemorepellent. And again, in line with what we found from the motility assay, the higher percentage of motile cells were actually observed at a greater depth. So in conclusion, 50 to 90% of cells in the pool water are motile, and a fraction of which are periodically motile. And this fraction of the periodic motile cells is generally consistent with depth. And the highest percentage of motile cells is not at the very surface, but at a greater depth. And these motile cells are indeed chemotactic to a moderate amount of nitrate, but higher concentration of nitrate might actually repel these cells. And with that, I'd like to thank everyone who have helped for this project, and I'll take any questions for now.
Any questions? What's the consistency of the sand? Like, was it sandy or silty?
It's silty.
Okay.
Yeah.
Yeah. So for like the periodic motile ones, could they have just been like coming from further away, kind of?
It could be. So I would say that the initial estimation what I give as a permanent motile cells is actually an underestimation of the cells, because they could be moving from further away, or it just takes some time for them to swim. But that's why I just count the total motile cells as just the maximum potential of motility for the sediment.
Hi everyone, thanks for being here. So this summer, and what I've been doing for the last year is I've been working on a device that can collect pore water in situ. And so being around just introduced us to pore water, and so I'm going to talk more about the chemical side of it. All right, so I'm sure you're familiar with climate change to some extent, but just the basics, there's a little power plant right there, and it's emitting our favorite greenhouse gas, carbon dioxide, and so there's a lot of it. The oceans are actually helping us out with this a lot, so they're absorbing about 1/3 of the atmospheric CO2 that we're putting out, which is good for us in a sense that It's making climate change effects a little bit more mild on land. So if this wasn't happening, we'd likely have more extreme storm events, more glacial melting, warmer temperatures, that kind of thing. And so it's good for us on land, but it does not come without a price to be paid by the ocean. And so this carbon dioxide mixes with the water to create carbonic acid. And so this is the stuff that's degrading the shells and breaking down corals and shells, and this has to do with coral bleaching as well as ocean warming. But this reaction is also in equilibrium, and so this is more of a short-term reaction because it's in the water column, and so it's in equilibrium with the atmosphere. So this goes back and forth. What I'm interested in more is these long-term reactions that happen in the sediments, so in the pore waters of the sediments. So this is how calcium carbonate dissolves. And so if you can imagine, we have our little critters up in the surface ocean, and they're making their shells, they're making their skeletons. When they die, their organic bodies degrade pretty quickly, and then the shells that were around them sink down to the bottom, and then they fall onto the sediment. And here's where they start to dissolve, thanks to this carbonic acid and this CO2 and H2O right here. And so they come into fairly innocuous parts, just calcium and bicarbonate. They don't do a whole lot, but this reaction right here, because it's a long-term reaction, it will start to sequester this CO2. And so after years and years and years, you will start to see some of the carbon. You'll start to see some of the carbon dioxide being sequestered from the atmosphere. But that does take thousands of years, and so we're trying to constrain it on a better time scale so we know more about it and what we can expect from that. So this is a pretty famous curve right here. This is the Keeling Curve. This is NOAA data out from the Mauna Loa station in Hawaii. So this is measuring carbon dioxide concentrations in the atmosphere. So as you can see, when they started measurements, it was about 320 ppm, and I think last I checked, we're at about 412. So yeah, this is easily available data on NOAA. So the red is atmospheric carbon dioxide, and then this green right here is seawater carbon dioxide. So once they started measuring that, it also lines up with the atmospheric carbon dioxide, and then this directly correlates to our ocean pH. So it's decreasing in pH. This is our ocean getting more acidic. So ocean acidification is happening. It's measurable. We can see it. So we already had a great introduction to pore water, but basically pore water is just the water in the pore spaces between the sediment grains. And it has a lot of interesting chemistry about, and also biology, as we just saw. But it can tell us more about the water and the sediments around it. So it's very valuable. It's hard to extract, or not extract per se, but it's hard to keep this sample in good enough condition that you can measure it and be confident with your results. So we are trying to extract it in a way that we can better measure our results and make conclusions from them. And so the typical way that we have done this, we'll see if this works, is, okay, the video's not working, sorry about that. But it goes down, it collects a sediment core, and then it comes back up to the ocean, and there's a boat, it comes onto the boat. Once it's on the boat, or you take it back home to the lab, then you can extract the pore water. And so commonly this is done with little filter sticks called rhizomes. They're very, very small filters, about 0.3 microns is I think the one that we use. And so then you extract the pore water up here. But the problem with this is while the water is still interacting with the mud from the bottom of the ocean to the top, there's crazy temperature and pressure differences. And so those differences will change the pore water chemistry while it's still in the mud. And so what we've done is we are taking out the pore water in situ while it's at the bottom of the ocean, so we don't have these changes in reactions. So, okay, it does not look like this video will work either, but here's a fixed animation of it. And so it goes down, and we collect it at different depths so we can get a profile, and these syringes will fire underwater. and then start to suck out the water from through these filter sticks, and so we don't get any mud in there, just water. Pretty amazing, actually. I think it's just like pure seawater or pour water once it comes back up. And so then it comes back up like this, and then we have our syringes full of pour water. It could not react with any sediment on the way up, so we don't have to worry about trying to correct for the changes that happen with the pressure and temperature on its way up. So this is what it actually looks like in real life. She's a beauty. So we did not make this whole thing. This is a multi-core, and so this frame was set for us. And then it's hard to see, but my syringes are up there, and the needles are down there, and that's sticking to the ocean. So that's what it looks like. And then, yeah, cool. I've shown you some nice cartoons. Does it actually work? You might be wondering. Mostly. There are some kinks that we're still working out, but for the most part, it's working kind of to what we'd expect. So this is some cruise data that we did at the end of July. We just went out to Spot and inserted it down there. And so, okay, so our green triangles is just the overlaying water right above the muds. That's kind of our starting point of the water that percolates into the sediment. The red is our needles, our new device that we're using, and the blue squares are the corings. That's the old method. And you can see there's a little bit different slopes in these two methods. So the core slope is a little bit steeper than our new device's slope. We're trying to figure out exactly what this means and which one is more reliable at this point, because we're still trying to, like I said, work out some kinks with our device. But they do line up somewhat together. So this is cruise data, and then here's a little bit of lab data, which always turns out nicer than field data, as I'm sure you're familiar with. So these blue squares are the traditional method. They're not quite the cores because it was just done in the lab. I just had a bucket of mud. I collected the bucket of the mud from SCMI down in the harbor. And so there are no temperature and pressure changes because it's been sitting in a bucket in the fume hood for this whole time. And so here's where we would expect the trends to line up between the new method and between the old method and our method. And they do pretty well. And so we're on this same trend right here, which we were happy to see about this because this is just done in the lab. And so we'd expect the same trend. So those are some preliminary results, and we are trying to go back out to sea in a few weeks to try and work this out some more, get some more data. So overall, we have built this device to sample pore water in situ, which reduces the artifacts associated with the temperature and pressure changes. With this, sorry, I didn't really talk about what we're measuring, but we're measuring carbonate chemistry parameters to learn more about how calcium carbonate dissolves on the seafloor and what this can do for climate change, and if this can help us on a human timescale, or likely it's much longer than that. But yeah, this is how we know more about it. Thank you. Questions? Yeah.
So you're measuring carbonate chemistry, so assume you're targeting sandy sediments?
No, we're mostly doing silty and clay sediments, yeah. So the next talk actually will be on sand. But yeah, so we're preparing to go out on a cruise in the deep Pacific, and so mostly mud, like clay.
Yeah, my question is for the device. So for the needles, so this device works for different sediment types.
Yeah. I have not tried it in sand, but I can't see any reason why it wouldn't work. And so it's I should have brought one to show you, but it has that little rhizome filter stick in it, and it's got a casing around it so that it can penetrate into the sediment without the filter getting torqued in any way. And it's connected to the syringe that pulls up. So I think it should work in sands too, but that's something we'd like to try.
And how deep does it go?
It can, right now, the deepest I can go is about 18 centimeters, which I would like to go deeper just to get more data. That'd be cooler, I think. But yeah, 18 is now.
Thank you.
Great, thank you.
Okay. weird thing on the screen. There we go. Okay, as she just said, I am Ashley. I'm in the chemistry department here at USC. I was on the Sinoski Summer Sustainability Fellowship this summer. And today I'm going to be talking to you guys about rhenium bipyridine catalysts with hydrogen bonding amines for CO2 reduction. And sorry that this is very chemistry heavy. So along the same note as what we just heard about, global CO2 emissions are a pretty big problem right now. Again, to sort of reiterate the point, we're now at over 400 ppm, and you can see this huge increase in the amount of CO2 just in the last, 70 or so years now. So this is a pretty big problem that we obviously need to find a way to tackle. So we're looking at the atmospheric side of this CO2 problem, and the main problem comes with CO2 being an incredibly stable molecule. So it's really hard once it's actually in the atmosphere to really do anything with it. It's linear in shape, so if you try to put in a single electron, take one out, whatever, it's sort of hard to get out of that linear shape, which makes it sort of just go in the atmosphere and sit there, which we obviously don't want. So just to sort of reiterate that here, we have CO2, you try to put a single electron in to make this anion radical, it takes a pretty negative potential of negative 1.9 volts. So just if you compare that to 0, that's a pretty negative amount that you have to put in there. However, what many people have been looking at more recently is doing catalysis, using some sort of molecular or other catalyst attached to a surface to sort of try to make this process easier. So you can see here, if you put in 2 electrons combined with 2 protons, you can actually reduce that potential down to negative 0.53, which is significantly less negative than what's required for just a single electron reduction. So again, the way that we do this is by using a catalyst. So chemists try to use nature a lot as inspiration. Obviously, this has been developing over millions of years. So in particular, we have CO dehydrogenase here, where we have these pendant amines of the residues, the amino acid residues here, that actually stabilize the CO2 adduct that forms during this catalytic process. So the idea is basically if CO dehydrogenase, this enzyme that's been developing over millions of years, is able to do this process very efficiently, very readily, it's a good idea to sort of emulate some of the themes that you find in these processes. So again, chemists have pretty frequently taken these themes as inspiration for their own work. This is just a select few examples of some of the catalysts that have been created for CO2 reduction. So generally, the idea is to reduce CO2 to CO, which you can then use that carbon monoxide. There's many processes out there that actually are used to turn CO into a variety of useful fuels, sort of as a renewable feedstock. So the problem is getting that CO2 to the CO. So these are just 4 examples of some catalysts that have been in the literature that actually very efficiently reduce CO2 to CO. You can see one common theme in all of these is that they have some sort of pendant proton. So like those amines here that stabilize that CO2 adduct during catalysis, we have OHs, amines, lots of amines. That basically, as you can see here, this example is actually from our lab. When you have that CO2 adduct during catalysis, if you have an amine, it can either directly or in our case indirectly stabilize that CO2 adduct that forms, which actually makes the CO2 reduction process much more efficient and energetically favorable. So these are more examples of catalysts with these pendant protons. This is more looking at a specific type of catalyst, which is these bipyridines. So 2 pyridine residues with either a rhenium or a manganese metal center. And again, in this case, they have pendant OH groups. In this case, also pendant OH groups, which as is shown by the example from our group, help stabilize that CO2 adduct during catalysis and really speed up that process. So our idea was to sort of combine these ideas with the bipyridine catalysts that are very prevalent in the literature. They're very well known for efficiently reducing CO2 to CO with the pendant amines that we've worked on in our group. So we wanted to look at what would happen if you have, first, more than one of those pendant protons available, and second, what happens if you remove the availability of those. So those were our four target complexes. We were able to make all four of them. This is just a quick crystal structure of them to show what they actually look like in the solid state. However, when they're in solution, which is how we actually do our electrochemistry, they do move around a little bit. So like in the case with the ones that have these pendant protons, they're sort of pointed inward right here toward that metal center, but they are able to rotate a little bit during catalysis. So the first thing we look at is the electrochemistry under nitrogen. So without adding any CO2, just to see what sort of features you observe. So one thing that we thought was really interesting is with the top two here, which are the ones with those pendant protons, compared to the bottom two, and this is just the parent complex, so that unsubstituted radium bipyridine. You can see that the two with those protons have this extra little weird feature in there. This is more what you expect from these complexes. You get a first single electron, generally pretty reversible reduction, followed by a second single electron irreversible reduction. However, this is very reproducible. We were seeing these weird little extra features here and kind of didn't really know what that meant. So one thing that we did to further study that was some variable scan rate studies, which basically is just ranging how fast you're scanning through these potentials. So you can see that when it's going really slow, which are these red ones, in the two with those pendant protons, you can see sort of the weird behavior with those extra features. However, when you speed it up, you don't see that anymore. You just see the two nice features that you typically observe. And essentially, we attributed that to some sort of chemical process that's occurring that is essentially too slow to be observed when you speed up the rate. And based on some literature precedent, we actually determined that this was likely some sort of hydrogen bonding interaction, where you can get two of the catalysts coming together and interacting through those hydrogen bonds, and actually can form some sort of dimerization, which is not ideal for catalysis. It sort of shuts off that pathway a little bit. But that's what is likely happening in those two versus more normal behavior in the ones without those pendant protons. So the next thing we needed to do was add CO2 to the equation. So what we're really looking for here is an increase in the current observed when we add CO2. Generally, that means when we're putting electrons and we're putting CO2 in, something's happening, ideally that CO2 to CO conversion. But the first thing, you just need to make sure that something is happening. So you can see through all of these, this is just with CO2, this is with CO2 and our proton source, which is trifluoroethanol. Generally, because as I showed in the equation, you need the electrons, but you also need those protons to efficiently promote this conversion. So these are basically just showing that when you add CO2 and you add your protons, you do get pretty substantial increases in current, which means that some sort of reductive process is occurring. We also look at what we call controlled potential electrolysis studies, which is basically you're holding the potential at a certain range for a period of time. You keep throwing electrons into that reaction, and then we actually take an injection of the headspace of the cell, put it in a GC, and we're able to see what we're producing. So in our case, we're looking for CO gas. So we hope that when we put it in the GC, we see a decent production of CO. Lots of numbers on this table, but the important parts are just the Faraday efficiency right here, basically. It's showing how much of the electrons we're putting in is being converted into CO. So unfortunately, these aren't the highest numbers. Ideally, you get pretty close to 100% for a process that's working the way you want it to. We sort of attributed that decrease in the efficiency and the amount of CO produced to that hydrogen bonding interaction we were seeing with those pentamines. However, we do get similar numbers for the two without those pendant protons also. However, they do still have the nitrogen, so they still can participate in hydrogen bonding interactions. And we think it's largely because those amines are so close to the metal center that they're just, when you have the CO2 adduct, they're really in close proximity where that's more likely to occur. So the next thing we wanted to look at was sort of a range of these catalysts. So these two, sorry, this is much easier to see on my computer. But the ones I've shaded out, these are two of the ones, the two symmetrical ones that I talked about that we've made and studied. This one was studied by another member of my group. So we essentially wanted to look at all of the other combinations, so moving the position on the pyridine ring. Again, with those dimethylamines, methylamines with a single pendant proton, and then actually looking at the ones with 2 protons on each side as well. So unfortunately, the ones I put in red here, we have not been able to synthesize. The synthetic conditions for these are pretty harsh, and after many attempts, we sort of decided to rule those 2 out and just go with these 4. So sort of adding to the range, comparing the dye-substituted versions with both 2 protons in each case. Again, this one we've studied in our group already, so 1 for each position, and then 1 with no protons to sort of compare having those versus not having them. And then I just have a little bit of preliminary electrochemical data. So again, this is the one that we've been able to crystallize so far. We need to grow crystals of these to actually be able to study them, so we know that they don't have any impurities in them. The other 3 have all been synthesized, but are in the process of growing crystals. So again, like the ones where we have that pendant proton in the other examples, we do see these 3 separate features, likely indicating that we're still having some sort of hydrogen bonding interaction. Interestingly, we don't see much of an increase in current under CO2. While this is not ideal, it's not incredibly uncommon because, again, sometimes you need those extra protons thrown into the reaction for it to actually go. So we were pleased to see that when we added that TFE, the acid source, we do see a pretty significant increase in current. So we are still getting likely CO2 reduction. We'll need to do those controlled potential electrolysis studies to make sure that we're actually producing CO, and then compare the amount that we're making to what we've seen before. So just a quick summary. We synthesized 4 new rhenium bipyridine catalysts with those pendant amines. And now, more recently, we're looking at the substituent effects and those hydrogen bonding interactions. And again, with our most recent one, we do see a current increase with the presence of acid. So for what I'll be doing next is work on crystallizing and studying the electrochemistry of the remaining complexes, and then again, comparing the amounts of CO that we produce with that first family of complexes that we made. So in acknowledgement, my professor Miranda has been a great help throughout all of this, along with both current and past group members. I also had an undergrad working for me this summer, which was really nice through all of these synthetic challenges that we dealt with. And then also Professor Ralph Hygas, who's been a great help with the crystal structures. And then these are my funding sources, most notably for the summer. The Sanofsky Sustainability Fellowship was a great help. And thank you all for your attention.
All right, so Jacqueline already presented, right? Okay, well, I was planning to go right after Jacqueline because it's on a very similar topic. So for those of you who don't know me, my name is Abby. I'm also a student in earth sciences here, and I'm also in the Barrelson lab, and I also study carbonate dissolution in sediments. So I'm at the risk of this being a total copy of Jacqueline's presentation, but there's a key difference. Jacqueline studies the deep ocean sediments, and I study this very thin area next to the continents called the continental shelf. That's the topic of my presentation. So can the continental shelf help buffer ocean acidification? So I understand that Jacqueline already went over kind of the basics of ocean acidification chemistry, so I'll skip all that. I just want to remind everyone that ocean acidification is not just an ocean, like deep ocean problem. This is it's global, it's happening everywhere, including off of the California coast. So these figures are from models that show that the water around California has been getting more acidic over the last 250 years. Do we have a power? Yeah. Okay. Over the last 250 years, and it will continue to get more acidic. So that's map view, and then these images are looking at the ocean from the side. So not only is the ocean getting more acidic at the surface, it's getting more acidic at the bottom of the ocean. So down here is where the water meets the sediment. So that interface is getting more acidic, and that's what I study, the chemistry that's happening right there. The big question is, are sands reacting to ocean acidification? And I say sands specifically because most of the continental shelf is sandy. So this image shows all the different types of sediments around the world, and Jacqueline is queen of most of the planet, all of this like pink clay. And then I'm left with this really pathetic these other colors right around the continents. So a very, very small area of the globe. And it's mostly sandy. And sands are different from clays because water can actually pump into the sands. So in clays, you have acidic water sitting on top of sand. And then it's just diffusion really working at that interface. But in the stuff I work in, you have water flowing, being pumped through sands all the time. So it's a very different chemical environment to think about. So the big question is, are sands reacting to ocean acidification, and specifically, are sands neutralizing ocean acidification? Are they neutralizing CO2? So again, I work on this like measly thing next to the continents. Jacqueline works out here. But in my defense, there is actually more carbonate globally in the shelves. So it's a very small area, but it has lots and lots of carbonate. And if you remember from Jacqueline's presentation, carbonate, when carbonate dissolves, it absorbs carbon dioxide. So there's the potential for the chemistry of the minerals here, the potential of carbonate to dissolve and sequester a lot of carbon dioxide. And we know, just thermodynamically, that when water gets more acidic, that carbonate will dissolve faster. So as the ocean gets more acidic, it's possible that the shelves start dissolving more, which is bad news that the shelves dissolve, but good news that you're soaking up carbon dioxide. Okay, so that's my big question. Do sands on the continental shelf absorb carbon dioxide? And the methods that I've used to study this include two main methods. Okay, I think this is, okay. Right. Two main methods, flow-through reactors and benthic chambers. And the approach I take with both of these is essentially the same. I pump water through sand, or I create a condition where water is pumped through sand. So in the flow-through reactors, there's water in these bags, and then the water is pumped up through the sand and then out. And then I measure the chemistry at the inlet, at intermediate points along the path, and then at the outlet. And I do this with both natural water, so just water that currently exists off of Catalina, and then acidic water, which simulates future ocean conditions. So I measure a bunch of ocean acidification related carbon fluxes, alkalinity, dissolved inorganic carbon, and also some carbon isotopes. And with these things, I can calculate how much carbonate is dissolving. So that's great. I can calculate it, but that's still a proxy, right? I'm not directly measuring carbonate dissolution. So then a second thing I do is I add C13 labeled carbonate to the experiments, and then I can directly measure how much of that isotopic carbonate is dissolved. So like with any good research, I have like multiple ways of measuring the same thing to verify the rates of dissolution. Okay, so this data is all from the sand columns that I showed. And those sand columns show very clearly that carbonate dissolution will increase as the ocean gets more acidic. That's not surprising, but it's nice to have numbers on it, and so that's what this research is about. So this figure shows, sorry, I made some changes to this, so I'm just gonna talk through it. The x-axis here is the saturation state of the water. So as the ocean gets more acidic, you move this way. So ocean acidification moves this way. We're currently here, and then by the end of the century, we'll be about here. The ocean's getting more acidic, and then the y-axis is the amount of carbonate dissolving. No carbonate dissolving, upwards to 70% of all of the dissolved inorganic carbon is from carbonate dissolution. This pattern is great. We see this trend moving upwards. There's a lot of scatter around here. So if I pulled all of these apart for my different treatments, I would see that flow rate matters, the type of sand matters. So Santa Monica sand has a different amount of carbonate, different amount of organic matter from Catalina sand. The methods matter, so the chambers are down here. Most of the sand columns are here. And then also the isotopic experiments show slightly lower rates. And I'm not going to get into that. There's reasons that the labels would dissolve more slowly. I can explain that later if anyone is super interested in isotopic labeling experiments. But the key findings are the carbonate dissolution will increase. I do definitely see dissolution happening at all saturation states. So that's interesting in and of itself, that we're oversaturated in current ocean conditions, but you still do see a little bit of saturation happening, a little bit of dissolution happening. So that's great to know that dissolution, well, I don't know if it's great, but it's good to be conclusive that dissolution will increase in the future as the ocean gets more acidic. However, that doesn't really tell us if the sands are net buffering or net acidifying, because there's multiple components of dissolved inorganic carbon. So there's CO2 coming from microbial dissolution or microbial respiration, which I'm sure, have you talked yet? I forget how the schedule goes. Okay, which is all related to Bing's work. So microbes respire CO2, which, as we know, is acidifying. So if I have sediment and I have CO2 coming out of it, but then I also have carbonate dissolution coming out of it, it's the balance of those two things that makes the sediment either a source of acid or a source of buffering capability. So this plot incorporates both of those things. So if I'm above the one line, think of that as more dissolution happening, and if I'm below the one line, you can think of that as more respiration happening. So we're here, there's currently more respiration happening than dissolution. So sands are currently making the situation worse. That's just naturally what they do. They accumulate a lot of organic matter, and then they pump carbon dioxide into the water. But as the ocean gets more acidic, we start moving up this line, and at some point later on in the second half of the century, we cross this line where sands actually start dissolving so much carbonate that they start buffering the overlying water. So that's really important for modeling and understanding what the pH of the coastal oceans will be. What's happening to the carbonate in the sand actually makes a big difference to the chemistry of the water. Okay, okay. So that's all I'm going to present today about the type of carbonate dissolution flux work I've been working on. But I started this summer working on some similar questions related to why we're seeing this much dissolution happening. The rates of dissolution are higher than we would have expected, and one reason that might be causing that are enzymes. So as much as I want to just be a geologist and not think about life at all, this is it seems that thinking about enzymes might actually help explain these data a little better. So this enzyme is called carbonic anhydrase. It's very common. It's in almost all cells, and it basically regulates carbon dioxide and the carbonic system. So it regulates CO2 to bicarbonate to carbonate, helps move those things around. So lab experiments have shown that this enzyme increases carbonate dissolution by orders of magnitude. Some recent work in the Barrelson lab has also shown, really interestingly, that this enzyme has to be attached to a mineral surface for the increased dissolution to happen. So you can't just have it floating around in the water changing water chemistry, it has to be attached. So that got me thinking, well, maybe in sands, that, if I'm seeing high rates of dissolution, maybe that enzyme is actually attached to the sand grains. So I did some experiments at Wrigley this summer where I measured the concentration or the activity of carbonic anhydrase in the water column and in the pore water. So already moving into the pore water, you see orders of magnitude more enzyme, which is not necessarily surprising. There's a lot more stuff in pore water than in the water column. But then moving from the pore water to the sand, so water I poured over sand directly, or just putting sand in my assay itself on a volume basis, there's orders of magnitude more enzyme attached to sand particles than there is even in the pore water. So I'll be doing more experiments with this using inhibitors and seeing if this enzyme actually makes a big difference in the field. So we know from these experiments that it makes a big difference in a bag in a lab, but in pore water, does this enzyme actually matter on an environmental level? Okay, so conclusions and next steps. First of all, conclusions, and I'm sorry this, okay, there's one more, sorry. I had a more updated version and I was afraid that the thank yous got left out, but they're here. Okay, so the main conclusions are carbonate sand dissolution will increase and is increasing in an acidifying ocean. And at some point, sands are going to transition from net source of acid to a net source of buffering. And the carbonic anhydrase activity is extremely high on sand grains, and it may be one of the reasons we see such rapid dissolution. I actually skipped over this because I didn't have time, but I mentioned that I used some chambers to measure fluxes, and those provided some really interesting results. But one of the main results is the nearshore is the forces in the nearshore are too rough, and so the signals were dampened because of leaking out of the bottom. So over the course of this fall, I'll be deploying some of those by scuba. And I'll also be doing more carbonic anhydrase assays. Okay, whew. Thank you are here. Thank you to everyone who has helped. A lot of you are in the room, everyone at the Wrigley Institute, and to the Victoria Burdick's Graduate Student Fellowship and Sandy Burdick's. Okay, any questions? Yeah.
Can you remind me what we talked about with like oysters contributing, or like mussel, like bivalve shells contributing to the cycle with carbon?
Yeah, so I don't remember exactly what we were talking about, but the inorganic carbon cycle is very counterintuitive. So some people, like there's been this idea that I've heard people talk about, like, oh, shells have carbon in carbonate. Why don't we just like grow a bunch of shells and bury the shells, and then that's carbon we're getting out of the atmosphere. But when you form carbonate, it's actually the opposite of dissolution, right? It's like you have carbonate and you dissolve it, or you have ions and you make carbonate. So when you make carbonate, you're actually producing carbon dioxide. So the net equation is you have two bicarbonates floating around in the water, and then you make a carbonate. One of those moles turns into carbonate and one mole turns into CO2. So growing a bunch of oysters.
So bivalve aquaculture would contribute to ocean acidification.
Yeah, through the inorganic carbon process. And then of course there's all like the respiration and the organic carbon that turns into inorganic carbon. That's another part of the equation too, which is also acidifying. So yeah, I would assume that on average, or on the net, the shellfish farming is a net,
Yeah. It's
forces
like,
the systems toward CO2.
There's like a lot of like bioremediation.
Yeah, for
points
nitrogen.
around that, but it's just in a different direction.
Yeah. Well, that's why doing like multi-trophic aquaculture is so great.
Right.
So,
Thanks.
yeah. Anyone else? Okay, thanks.
Hey guys, as Linda mentioned, I'm going to talk to you about heterotrophic nitrogen fixation associated with decomposing macroalgae. Before I dive into that, why do we care about nitrogen? Nitrogen is an important constituent of many of life's building blocks. So from the smallest organism to the largest organism, everyone needs nitrogen to build biomass. Some people have already talked about this, like in protein synthesis. In the marine setting, it's often thought that nitrogen is a prominent limiting macronutrient for marine productivity, including macroalgal productivity. So where do macroalgae usually get their nitrogen and other nutrients from? They usually get it from the water, or it's typically thought that they get it from the water column, where it can accumulate via nutrient regeneration from the underlying sediments. You can also have seasonal upwelling events that bring up deeper nutrient-rich waters that bring up nitrate, phosphate, etc. And then because a lot of these macroalgal sites are coastal, you can also have nitrogen loading events through eutrophication, etc. There's also another one that I study, is biological nitrogen fixation, which is the process of converting dinitrogen gas into a bioavailable form of nitrogen that's carried out by a specialized group of prokaryotes known as diazotrophs. They use this enzyme called nitrogenase to catalyze that reaction. Typically in the macroalgal realm, even though there's been a few studies that show that macroalgae partner with diazotrophs and they can contribute to the nitrogen budget for macroalgae, biological nitrogen fixation is often overlooked as a source of nitrogen in macroalgal systems. That's not the scope of this talk. That's with living macroalgal systems. I'm talking about decomposing systems for this talk. All right. However, there is such a thing as too much nitrogen too. Sorry, the picture resolution is kind of grainy. I just pulled these off the web. But what happens when macroalgae have access to too much nutrients, such as nitrogen, is you can have massive bloom events of macroalgae, such as this golden tide that's depicted here. This is Sargassum that ended up in the Caribbean, I believe. And basically, these can be really detrimental to the surrounding system when you have this massive bloom, because it can lead to coastal anoxia, hypoxia, and also death of the underlying benthic community. Well, you can also have bloom events with green macroalgae. This one is the Ulva species, and I think this actually caused, it's a really good example of how it not only has detrimental impacts to the ecosystem, but it can also be really costly for economies to clean up. The green tide that happened in China during the Olympics was like a perfect example of how that was really, really costly. These are, however, examples of extreme macroalgal productivity, but even with the decomposition of really small or regular amounts of macroalgal biomass, it makes sense to think that as microbes degrade that biomass, you're going to release all the nutrients and organic matter that was once locked up in the macroalgal tissue. So in these detrital systems, you would not expect nitrogen to be limiting. However, there have been some studies that suggest that for the microbial community associated with breaking down the macroalgal biomass, nitrogen actually can become limiting, usually thought to be later on in the decomposition process. So this got me interested in the potential role that nitrogen fixers may have with decomposition, because they have their own special reserve of nitrogen, right? They can supply their own nitrogen. So I'm interested to see if they play a role in the breakdown of macroalgal detritus. So what I've done this summer and in previous summers is gone out via freediving and collect substrate-bound macroalgae, and then I bring that back into lab, put the biomass into 200 micron mesh bags, and allow them to degrade and continuous flow through seawater tanks out in Catalina for X number of days, anywhere ranging from 20 to 30. It all depends on how quickly that biomass is degrading or decomposing. And then none of this work would be possible without the help of the REUs that have been working with me in the past few summers. What we do then is I subsample the aging seaweed detritus every few days, and then I carry out what's called the acetylene reduction assays. For the sake of time, I'm not going to dive into the methodology too much. Suffice to say, I use a gas chromatograph to indirectly quantify nitrogenase activity. Nitrogenase, reminder, is the enzyme that's responsible for nitrogen fixation. So I'm just going to go into a few of the results that we have. So on this slide, this was a decomposition experiment that I did with 5 different species of macroalgae. 2 brown macroalgae, Cysticerra and Dictyota, 1 green Codium, and then 2 red macroalgae, Sphaerogopsis and Plecanum. They're the ones that are smaller up in panel D and E. So there's a couple of different trends I want to point out with this figure. I'll start with the brown macroalgae. You'll notice with both Cysticerra and Dictyota, they're not copy and paste graphs, by the way. They just happen to show very similar trends. You'll notice in the beginning, you have very low to below detection limits of nitrogen fixation rates, but then after just 5 days of decomposition, you get this drastic increase in nitrogenase activity, which is then sustained for a number of days before nitrogen fixation rates begin to decrease. And the other thing I like to point out is these incubations are done in parallel with dark and light incubations, and you'll notice the dark is represented by the darker colored, and the light is represented by the lighter. Dark incubation, dark nitrogen fixation rates are comparable, if not higher than the light nitrogen fixation rates. And in the absence of light, we're inferring this dark nitrogenase activity to be the result of heterotrophic nitrogen fixation. The trend is not as pronounced with Codium on panel C here, but it's somewhat similar, right? Lower in the beginning, you have an increase that's sustained for a number of days before you get to rates that are below detection limit. The red macroalgae, if you notice the y-axis is a different scale. It's going from 0 to 3 as opposed to 0 to 200. So it's over 100-fold lower. So it's hard to say anything more concrete about what's happening with the red macroalgae decomposition and the role that diazotropes are playing in that system. So this summer we actually redid the decomposition experiment with Codium, and now we got a much cleaner looking result. And this was due to increased sampling frequency throughout the decomposition process. What you can see is there is actually an initial increase going from day 0 to day 2, but that lower nitrogen fixation activity in the beginning is sustained for a while, and then around day 10 you get this drastic increase in nitrogenase activity again, sustained for a number of days, and then around day 32 is when you start actually seeing that decrease in nitrogenase activity. So it turns out we're actually seeing the same pattern with Codium when we redid the experiment in a much cleaner setting. And again, the darker bars represent dark incubations, and you can see the importance of heterotrophic nitrogen fixation. It's actually a little bit more important than light incubations in this system. Okay, I also wanted to talk about another species that we were able to sample, Ulva, which was the one that was responsible for the green tide in China that I mentioned in the beginning intro. If we just ignore the red boxes for a second here and the blue bar graphs, what happened is if you just look at the green, on the left side is the dark incubation panel, and the light side, on the right side is the light incubation. You see a similar trend where in the beginning you have lower nitrogenous activity, increases, and then it's sustained for a while before the rates start to decrease over time. Interestingly enough, before I was doing anything with Ulva, there was a paper that was published in 2015 where during one of those green tide macroalgal bloom events with Ulva, these researchers, Zhang et al., subsampled the detritus and they sequenced a bunch of it to see what kind of microbes were associated with it. And one phylotype, the Desulfovibrio-related phylotypes, were found to dominate the heterotrophic diazotrophic community. Desulfovibrio is a sulfate-reducing microorganism that can also fix nitrogen, or is thought to potentially fix nitrogen. So I was interested in doing sodium molybdate inhibition assays. Sodium molybdate inhibition assays are represented by the blue bar graphs. Sodium molybdate is known to be an inhibitor of sulfate reducers. So theoretically, if sulfate reducers are present in the diazotrophic community in the amendments with sodium molybdate, you might expect to have lower nitrogenase activity. And what's circled in the red box here is you actually see drastic inhibition when you put sodium molybdate in these parallel assays. So it kind of confirms what they were seeing with sequence data, that we do have sulfate reducers being a prominent portion of the diazotrophic community with Ulva. And actually, interestingly, in the past, when I've done the decomposition experiments with brown macroalgae, we've also seen similar things when we add sodium molybdate. With both Cysticerra and Dictyota, you see reproducible instances where sodium molybdate can really inhibit nitrogen fixation activity, suggesting again that sulfate reducers may be an important part of the heterotrophic decomposition or heterotrophic nitrogen fixation associated with decomposing macroalgae. Few concluding remarks. It seems that there's higher dark nitrogen fixation rates with senescing brown and green macroalgae, which suggests that heterotrophic nitrogen fixation may be really important in these detrital systems. Surprisingly, there's drastically lower nitrogenase activity observed with the 2 species of red macroalgae, so a little bit more work with reds is going to be needed in order to get a more clear understanding of what's happening in that system. And then lastly, sulfate reducers are an important member of the Diazotrophic Macroalgal Association. A few references, and of course none of this work would be possible without the Wrigley Fellowship, everyone at WMSC, especially Lauren and Kelly for always accommodating us and helping us out. And then all the REUs in the past and also present members of the Capone Lab, Catherine and other fellows that have helped me collect macroalgae on multiple occasions, and then Linda, thank you for coming all the way over here for this. And then I just threw a bunch of stuff, so if you're like Jack at all, if you got any questions, I'll take them. I think I have a few minutes. So.
Any questions? Abby?
This is potentially not related, but Vibrio is also like a pathogen, right?
Yeah.
Is it the same Vibrio or?
No, this is disulfovibrio.
Okay.
So not like you're thinking of like Vibrio without the disulfo in front of it. This one is a sulfate reducer. Yeah.
Okay, so I was just wondering if there might be any more rotting algae
leading to pathogenic Vibrio. Not that I'm aware of. No. Yeah.
Was this before or after he was locked in the bathroom?
After he was locked in the bathroom. All right. Thank you guys.
Okay, great. All right, good afternoon, everybody. All right, good. So my name's Anne. I'm Moss Landing Marine Labs, which is all the way up in Monterey Bay. But I've been coming down here for about 14, 15 months every month to sample and explore Catalina Island. So I'm going to share with you some of the changes and interesting things that have been going on in our kelp forests out there. So we've been talking a lot about a lot of the little microscopic, really interesting interactions that are happening on the seafloor and the sand, or out on the sandy bottom with the sharks. But this is what it's really all about, is the foundation species, giant brown kelp. It's a seaweed that provides habitat, it provides food, it mitigates physical ocean conditions as well as the chemical ocean conditions of the environment. And this is actually what Catalina used to look like several years ago before we had the warm blob and the El Nino. It was this towering city of kelp that provided homes for fish and vertebrates, as well as some very enthusiastic humans who would prefer to live underwater. During the course of the El Nino and the warm blob, it went from looking like a kelp forest to looking like this, to looking like this. So we went from this thick, dense kelp forest community to this dramatic change of having a Sargassum forest or thicket, depending on how you'd like to call it. And that changes not only the seaweed community, but it then influences all of the animals and people who then rely on this community and live and work around this community. And so, having been on the island when this occurred and also being very interested in ecology, I was really curious as to why Macrocystis, the giant brown kelp, has been so slow to come back and why this particular type of Sargassum has been so successful. Catalina is a really interesting place in that it has had multiple invasive species over the years. It has had multiple native species of Sargassum, but this particular one seemed to just spring up out of nowhere and take over everything. And so kind of starting at the beginning of things, I got interested in the life histories of these species. Macrocystis is a perennial, so it can grow, survive, reproduce all year round, whereas the Sargassum is an annual with an animalistic or more plant life history. Now, what does that mean? This is the life history of a giant brown kelp. The big piece we see is a sporophyte. It has its two ends, so it has both genetic materials, just like we have from its parents. It produces zoospores, and those zoospores will settle on the substrate of the reef and develop either egg or sperm, and then those then find each other using hormones to make a tiny sporophyte, which will then grow again into this big, beautiful kelp plant. It takes time, it takes proximity, it also takes specific environmental conditions. They found in Youth 2019 that even if you have all of the nutrients and all of the light available, if it is too warm, the kelp will not go through this phase of producing zoospores. So that's one thing to keep in mind throughout this talk. And then we have Sargassum. So Sargassum, like weedy plants you might see on the side of the road, has one mission. It's to live fast, reproduce fast, and die fast. And so because of that, it has a very different strategy. Instead of producing spores and then have to go find each other to make a new plant, instead everything happens right on the adult individual. If the adult's growing and able to reproduce, it must be a great spot to grow. And so why wouldn't it be great for its babies or zygotes to grow there? And so it produces these little nodules on the adult plant that have little pits in them called conceptacles. And in those conceptacles, there's male and female reproductive structures. Ideally, it gets fertilized by a different plant, but if it doesn't find another individual, it will self-reproduce and then drop these little growing zygotes onto the reef floor that immediately can then spring up and start the whole cycle over again if conditions are, again, great for growing. So, based on this knowledge of life histories and some of the things going on Catalina and in the Southern California Bight in general, I wanted to take some time and explore what is happening in these kelp forests. We saw all the kelp go away, all the sargassum come in, and it was so bad that it was even in newspapers talking about the devil's weed and will the fish ever come back and all these things. So in a lot of El Niños that we've had in this area, it usually takes 3 to 4 years for the kelp to come back. So I wanted to see, well, is the kelp coming back, or are we in some kind of new, strange ecology cycle? And to do that, I did monthly surveys to look at the proportion of adults, juveniles, looking at their biomass on the reef, as well as what is the proportion of reproduction and how there could be competition between these two very different strategies playing out on our reefs. And so I did a bunch of field surveys, and then I also did some laboratory work to explore these ideas. All right, so first couple questions about juveniles and adults and what's happening out there on the reef. So starting in June 2018, I started identifying areas to set up a bunch of transects to survey for the kelp and the sargassum. And so using knowledge of the various Wrigley staff and other locals on the island, I found places that had historically had nice large dense kelp forests, areas that had been studied in other papers looking at El Nino recovery and the sargassum invasion, and trying to figure out, okay, so what are the density of kelp and sargassum over the course of this year, and how is that going to be changing through time, as well as how does their reproductivity and fecundity change over time with those oceanic conditions. And so I found 4 sites, and at all 4 of those sites, I sampled monthly, and I set up kind of this matrix of sampling within the kelp forest. Each site had 3 10-meter transects taken, and each of those had a 2-meter by 10-meter kelp swath where divers went down and counted every kelp within that 2 by 10, looking at the stipe counts to identify biomass, looking at the number of juveniles. If they were lower than a meter, where you typically take that stipe counts. And then for the sargassum, because when they're big, they're easy to count, but when they're little, you can have 50 to 100 in a meter. So we did some quadrat sampling for those rather than trying to count every single sargassum in a 2-meter by 10-meter swath. And then also adults were collected in the case of sargassum and measured in the case of kelp to figure out what the average biomass of these individuals were. Now, the reason that we didn't collect the kelp was because it is kind of a valuable individual out there. We want our kelp to stay in the water. We want it to reproduce. It's also rather large. You could, in theory, collect only 1 kelp plant, and it would fill up your boat, and both the staff would be very unhappy with all the kelp slime on the boat, and also I'd be very unhappy having to take the boat out multiple times to get 1 kelp plant. So instead, I kayaked around the around various places of the island and collected drift fronds that were attached to holdfasts to make an equation following Dan Reed's methods from 2009 to figure out if I measure how long and how many fronds a kelp has, can I figure out how much it weighs? And so using those equations and collecting a little over 200 kelps from the drift, I was able to create this regression last summer, which now allows me to take all the data I was collecting on scuba and calculate the biomass of these kelp forests. which looked something like this. So these are some of the pictures of what was going on underneath. In some months, the kelp was really big, lots to count and calculate. Some months, there were just little shrubs of sporophylls pumping out zoospores. So there's some very interesting and strange things happening out there. Sometimes the sargassum was smaller than the palm of my hand, other times it was bigger than me. So there's a lot of very interesting interactions, and I'm currently putting together the graphs that kind of explain and show the time difference of when things are at their biggest and healthiest versus when they're all little babies and cute little seaweed on the reef. So right now I'm crunching the numbers for that to compare all of the biomass, as well as analyzing some microscope photos of those tissues to pinpoint exactly when the kelp versus the sargassum is most reproductive. Because if you think about it, sargassum, looking at those life history diagrams from earlier, already has a bit of a head start with its timing, and so if it's dropping zygotes before the kelp even has a chance to settle on the reef, that could be driving some of the dynamics we are seeing out there on the island. All right. My other question was kind of looking forward as to how our temperature and chemistry kind of playing into the success of the microscopic baby life stages of these seaweed. And so while I was out there during the spring, which seems to be when the two species are the most reproductive, I was collecting reproductive parts, so sporophyll reproductive blades from adult kelp, as well as the reproductive sargassum, and bringing them back to the lab and growing them under cool temperatures, normal temperatures, and projected hot temperatures with high and low nutrients to mimic what ocean conditions have been like and could be like again if we get another strong El Nino in the near future, to try and see, okay, does the presence of sargassum itself cause any of the issues we might see with kelp getting a start, or is it purely driven by that temperature and nutrient dynamic? And with that, what it looks like is kind of like this. So over here on the far side of the screen, those are some little kelp gametophytes. And as they grow, they elongate, they start producing the oogonia and the anthrazoids, but they quickly can get overwhelmed by these sargassum zygotes. The little dots you can kind of see behind this lower one are the kelp gametophytes just entirely getting overshaded. Interestingly, they all seem to do a little bit better on warmer temperatures. I also had some kelp gametophytes from Monterey, which did not like the warmer temperatures, so there could be some local adaptations going on with the Catalina kelp where it is starting to figure out a way that to meet those warmer temperatures and maybe combat things over time. However, it does require time for us to get that, and so we'll have to see as climate change happens whether or not there is enough time for the kelp to fully kind of punch back at those invasive species in higher temperatures. All right, so with that, I'm still on doing a bunch of photo analysis to pinpoint exactly where that switch seems to happen between cool and warm. So stay tuned. But I'd like to give a big thank you to all of the Wrigley Lab support staff, to Linda and the folks who allowed me to dink around on the island for a year and a half almost, and the other Wrigley fellows who went diving with me, helped collect things for me, all kinds of things could not have done without you, and the rest of my amazing volunteers, Mike and Diana, for helping me kind of work out some of the kinks, especially with my laboratory things. The rest of my beer pigs, which is what the Moss Landing Phycology Lab group is called. And yeah, again, all the volunteers and people, it's definitely takes a team to get science off the ground. So with that, any questions? And thank you for your time.
Okay, my name is Lauren Smith, and I'm a PhD candidate at UCLA, and I'm presenting on some of my dissertation work that I worked on this summer, evaluating the role of environmental drivers that may facilitate expansion of the invasive macroalga Sargassum horneri. So this is my study species, and it is native to Japan and Korea. It first arrived in Southern California in 2003 in Long Beach Harbor. And likely it was introduced there via a shipping vessel. Since then, the next 3 years, it was found in 3 locations on Santa Catalina Island. And since then, it has spread over 750 kilometers from Santa Barbara to Isla Natividad, and there have actually now been reports from both Monterey Bay and Santa Cruz. So likely farther than this, although I don't think those are established populations yet. Anne already did a really great job of introducing Sargassum honorae, so I'll breeze through my slides about that. But it is an annual species, as she said, so it lives its entire life cycle in 1 year. Generally speaking, that life cycle starts right about now, so in the summertime, it is in its smallest stage. In the winter, it becomes reproductive, and then in the spring, it starts to senesce. It grows to be about 3 meters in height. It is highly fecund, which makes it particularly well suited to invade. And as Anne said, it is capable of self-fertilization, which is another great invasion technique. There's a lot of concern that Sargassum honorae is spreading into areas that we have historically considered dominated by kelp forests. That's okay. Our Southern California kelp forests are characterized by cold, nutrient-rich waters. The dominant species is Macrocystis pyrifera, which is perennial, so it lives over several years. It grows to be about 3 meters in height, so just like habitat-wise, it is a very different structure than Sargassum honorae. And most importantly to my research, it starts to decline at and above 20 degrees Celsius. Which brings me to my question, do temperature excursions facilitate expansion by promoting the growth of Sargassum honorae? So for the next few slides, I'll talk about why I'm asking this question, where I conducted this research, and how I conducted this research. So a lot of this starts with the El Nino Southern Oscillation, which Anne gave us a little bit of an intro to. What happens during the El Ninos, we have a warming of our ocean surface to above average temperatures in the Pacific Ocean. The speed and strength of the currents change. The health of coastal fisheries are impacted. The local weathers experience a reversal, and that upwelling I discussed that usually brings that cold, nutrient-rich water is reversed. So we have downwelling, so we have warm, nutrient-poor water. In particular, this question was driven by the 2015-2016 El Nino because it was stronger with prolonged temperature increase. You can see it right there at the end. And I'll zoom in. You can tell from this graph that the 1987 one is quite a bit lower. This one actually goes off the graph. And so it's a very strong and prolonged El Nino, and previous research on Catalina Island found that Sargassum was more abundant post-El Nino than pre-El Nino. In a lot of places, that coincided with a decrease in kelp, and as I said, an increase in Sargassum honorae. So I conducted my research on Santa Catalina Island. In the west side of Two Harbors, a place called Isthmus Point. And as I said, Sargassum has been particularly prevalent on Catalina. In some locations, it has become the primary placeholder. And so I'm doing two kind of studies, one in the field and then one in the lab. So I'll discuss both of those here today. In the field, I am collecting Sargassum honorae, attaching it to ropes, and then putting it back into the water to measure growth over time. I'm doing this with 3 size classes, less than 10 centimeters, between 10 and 100 centimeters, and greater than 100 centimeters as available. As I said, they're annual, so hopefully I'll be able to get all 3 species at once, or all 3 sizes at once in the spring, but not over the summer. They're going to be deployed in the field for 5 days with temperature loggers. I plan on doing this twice monthly, so I started in July, and then I'm going to compare the percent growth with the average daily maximum temperatures. And kind of just going into what exactly that looks like, my dive buddy Emily and I collect Sargassum honorae. We bring it back to Wrigley, where we put it in little pantyhose to keep it safe. And then we want to get a consistent wet weight, so we put it through a salad spinner. So for 1 minute we spin algae salads, and then we go ahead and we weigh and measure all the individuals, and then affix them to the rope and put that rope back into the field. So that's what this looked like. So you can see my rope right here, and I have a Sargassum there, a Sargassum here. And I did that twice in July, but didn't go as planned, which is okay. So this is the very first time we did it. So this is the percent change in weight, and this is the percent change in height. The temperature during the peak hours of the day, so I did like 1 to 4 p.m. was 19 degrees Celsius. And these, I was able to get 2 size classes, so small is in green and medium is in blue. And as you can see, they didn't grow at all. And so kind of took a step back and looked at what I thought happened. And looking at the algae, it looked like it had been predated on. And so if I'm just interested in looking at temperature, then I probably don't care if it gets eaten, because that might be impacting the signal of temperature if the whole time they're just being eaten and they're not growing. So I kind of thought about what I wanted to ask, and I decided I want to look at both things, both temperature and predation and together. So I updated my methods and I jailed half of my algae. And so put together cages that are closed on the top and closed on the bottom. And what I found from Emily's research last summer is when you give algae new platforms on the bottom, they no longer compete with the algae surrounding them, and so it gives them a new kind of habitat to grow in. So I put the other half on platforms. This is kind of what that looked like. These are Emily's cages, so they're not closed, but put them on lines out in the ocean. I did that once in August. And it was difficult because the platforms and the cages were next to each other, and the cages pulled the algae off the platforms, and all of the algae died. But we looked at the algae in the field, and it was also dying. And so looking at the conditions, the water had been really turbid and full of probably plankton, and so the light conditions were pretty poor, and so we think that that's what happened. But Emily and I just finished the second round on Tuesday, and so I have my first set of data. I haven't really analyzed it yet, but the uncaged, these are uncaged and these are caged. Mediums did the best in change in weight. Our error bars are still kind of big on some of them. So still trying to figure that out and haven't really added temperature into the equation yet, but planning on going out next week to do our second round. Which brings me to my future plans. I plan on continuing these field surveys on Catalina Island, again trying to capture a wide range of temperatures. And then I'm also planning on conducting a mesocosm experiment in February when the water temperature is a little bit colder. And the way that's going to look is I'm going to collect 30 small sargassum, put them in jars, and I'm going to use aquarium heaters to set the jars to range from between 15 and 25 degrees Celsius, again for 5 days. And then compare the percent growth with the average temperature. I did a preliminary study with just 9 jars last summer, and I did find that Sargassum horneri increased with increasing temperature in the lab, so I'm hoping to replicate that and show that in the field as well. With that, I'd like to thank my funding sources, the Wrigley Institute for the Fellowship, UCLA, Santa Monica Bay Audubon Society, and LeCret Center, as well as my advisor, Dr. Peggy Fong, the USC Wrigley staff, my dive buddy, Emily Reisner, and the rest of the Fong lab. Any questions?
So why do you think the uncaged one did better this time?
I'm not sure. I think some of the cages, they grow stuff on them, and so I'm trying to do it for a small amount of time that that's not hopefully impacting it, but I haven't really dug into it yet. So I need to look at my notes and see if maybe that's what happened, or if maybe the bigger ones are big enough that they're hitting the sides of the cages, and so they're losing mass that way, or if something else is going on.
All right. Good afternoon. My name is Juan Pablo de los Rios. I'm a second-year graduate student in the FISA group, and today I'll be presenting my summer research on developing rare earth catalysts in order to tackle the environmental plastic buildup. So, as you know, plastic pollution is it's a huge problem today. The World Economic Forum published this figure in 2014, demonstrating that in that year there were approximately 78 million tons of packaging plastic produced. Out of this 78 million tons, 32% leaked into the oceans or the environment, 40% ended up in landfills, 14% were incinerated or processed for energy recovery, And out of 78 million tons, only 14% were collected for recycling. This 14% though does not indicate that all the plastic products were effectively recycled. As a matter of fact, 4% of the 78 million tons were process losses. What this means is that plastic products are lost during transportation or falling off of the conveyor belt. 8% were subjected to cascaded recycling, and this method indicates that a product is recycled into a low valuable product, such as a water bottle being turned into carpet fibers. And out of this 78 million tons, only 2% were collected for a closed loop recycling method, which simply means that a water bottle ended up as a water bottle to be used again. So this is clearly a problem, and the FISA group is trying to address this problem of environmental pollution. So how exactly are we trying to do this? Well, the FISA group has the idea of designing catalysts that can first make degradable plastics as replacements. And pictured here is polylactic acid, which is currently in the market. And your corn cup right here is a 100% bio-based product that is commercially available. And our second strategy is to degrade plastics accumulating in our oceans and environment. And the perfect example that I can give here is PET, which is a major component in water bottles, and it's one of the more complicated plastics to degrade. So I will only be talking about our first strategy here because that's what my summer research was focused on. And how exactly do we want to make these degradable plastics as replacements? Pictured here is the copolymerization of epoxides, which is your chemical species here on the red, and your cyclic anhydrides, which is your structure here in the blue. And by using a catalyst, you copolymerize these two monomers to form your polymer or plastic. So the wonderful part about this transformation is that it has a wide range of monomers. There have been over 20 epoxides reported as monomers, with those in black being the ones coming from petroleum, and your green are often biosources, or they come from biosources. And likewise, there have been over 20 reported anhydrides, and they can be cyclic, bicyclic, or tricyclic. What I want you to know is the different R groups on these anhydrides and epoxides, so they can have different functionalities, the monomers that ultimately give different properties to the polyester or polymer. So overall, there are over 400 possible polyester structures that we can develop with varying thermal properties. For example, glass transition temperatures ranging from -44 to 184 degrees Celsius. And like I said, the different functional groups on your plastic could be modified after polymerization to yield plastics with even different properties. And the overall scope, as I said, is to make degradable and biocompatible materials based on our catalyst here. So as my title indicated, we're focused on using rare earth metals. Picture here is your periodic table. You have scandium, yttrium, and then your lanthanide series, which are also referred to as rare earth metals. The wonderful part about the rare earth elements is that you have a very subtle difference in ionic radii as you transgress from left to right on your series. This is a unique trend that is not observable anywhere else in the periodic table. So by selecting a specific metal, you can vary the steric crowding of your catalyst that ultimately gives different reactivity to it. I know you may be wondering why rare earth metals. Actually, they're not as rare as the name indicates. Picture in red here, they're very small, but you have your nickel, copper, and zinc, which are your major industrial elements. And pictured here on the blue are your rare earth metals, which are honestly not that far off from your common industrial metals. And then on the green, you have your precious metal. Your gold is here, your silver, your platinum also. So these elements are actually heavily used in industry and are not rare whatsoever. So the Pfizer group wants to use these metals to make our catalyst, right? And these are the structures proposed that we want to synthesize. Every colored letter indicates a variable that can be differentiated or could be selected that would give your catalyst different reactivity, varying your steric crowding or your electronic properties. Given the time, and since I've been only working on this project for a small amount of time, I will indicate only talking about the first structure here, which are referenced as your beta diketaminate ligands. They're interchangeable with the name NAC-NAC ligands, but in this presentation they've been abbreviated to BDI ligands. Your R groups here could be varied to be either symmetric or asymmetric ligands. You can kind of see the structure, how it could be symmetric or asymmetric depending on the substituents that you choose on your backbone ligand framework. So, during my summer research, I was able to effectively synthesize, characterize, and optimize two particular ligands, BDI1 and BDI3. As you can see, they're symmetric with their respective phenyl groups for BDI number 1, and this 2,6-isopropyl phenyl on the backbone. BDI number 2, my data demonstrates that it's been synthesized, but I'm currently working to washing it and optimizing its yield. So it's not yet pictured, but hopefully in the upcoming weeks, I'll be able to have it on future slides. As I said, the asymmetric ligands are also possible to do. They're a little bit more complicated to synthesize given that it's a two-step reaction. I was able to effectively synthesize intermediate, and there's current investigations undergoing using p-tosulfonic acid at 120 degrees Celsius in toluene with different anilines, either your aniline or your 2,6-dimethyl aniline, to produce your BDI ligands, your asymmetric ones. They are pictured in red because, as I said, I have not been able to characterize them yet, but we're very hopeful of this reaction, and we're manipulating temperature and solvents to see how these structures can be obtainable. Likewise, I was able to synthesize another asymmetric intermediate. In this case, I have a tert-butyl group. This reaction, it would be using a microwave reactor at 150 degrees Celsius for about an hour. So it's actually a quick reaction. I don't have to add anything else into the microwave flask. Unfortunately, the one that we had access to, it was only a temporary microwave, so we had to send it back. For the group that we were borrowing it from, had to send it back. And now that we have access to one here at USC, I'll be exploring in the upcoming months this reaction using aniline and 2,6-diisopropyl aniline to produce asymmetric BDI ligand 6 and asymmetric ligand BDI 7. During the summer, I was also able to do some metallation reactions with my BDI number 3. Apparently, this is the one that is used mostly in literature, so I had a little bit more knowledge on what reactions I could do to develop my catalyst. And in this case, what I'm trying to do is deprotonate or remove this proton right here to make a negative charged species that can ultimately be reacted with a metal and thus form my catalyst. My data shows a lot of promise on this structure here because the signal that you start with on BDI number 3 is actually not observed for my species here. So I'm very hopeful for this reaction. I will continue to investigate it and hopefully obtain nicer yields with my deprotonated ligand. I will also be attempting these reactions here now that I know my species can be deprotonated with the trimethylsilyl here. Once I have my lanthanide precursor with the respective ligands that can deprotonate my BDI number 3, I would basically just be able to do an exchange and form my metal catalyst, the first one that I've made yet. So I'll be exploring this reaction in the upcoming months to develop my first catalyst. So just an overview and future directions. My VDIs here are the ones that have been effectively synthesized. The ones on red here are the current ones that I'm trying to develop. And like I said, this reaction is probably the one that I'll be running first, just to see if I can obtain the first catalyst, because it's been a series of months that I really want to obtain a metal structure. And from there, obviously the path is really open to the other VDIs that I synthesized, just burying these R groups here, right? So once I synthesize all these complexes, they can be subjected to this reaction here to hopefully form even more catalysts that can then be tested for the co-polymerization. The metals that I'll be looking at first will be yttrium and lanthanum. So we're very hopeful about our work, and we'll see in the upcoming months where these leads. I would personally like to acknowledge my whole group, Professor Megan Fieser, my lab mates Yvonne and Sophia, the undergrads at our group, Colette and Alexi, and our visiting student, Kelsey. She's not pictured here. She had to return to university. And I would like to personally thank Norma and Gerald Sonosky for funding my research this summer and making our sustainable chemistry go forward. I don't have a question slides, but you're more than welcome to ask a few questions.
Questions? There, go over there.
Yes.
What instrumentation, or how do you know what kind of molecule you get after you synthesize it? How do you know?
So organics, the ligands that I'm trying to do, we use nuclear magnetic spectroscopy, nuclear magnetic resonance spectroscopy, and you just detect the signals of the protons and kind of build the structure depending on your signals. We're also hoping to do some elemental analysis which gives you the percentage of each component that you have to kind of back you up that you have developed that structure. Yes?
What are the benefits for having like a symmetrical versus an asymmetrical catalyst?
That's a very good question. So we're just trying to play with the whole sterics at first of the catalyst. So having different groups will kind of give us an insight of the best rates for the polymerization, and maybe the symmetric ones work better with some monomers, the asymmetric work better with other monomers. So we're kind of just exploring the whole spectrum. Thank you.
So my name's Emily. I'm a PhD student at UCLA, and I don't know if you guys heard about the alga Sargassum horneri, but I'm also going to be talking about it. And some of my research this summer, and kind of continuing on from last summer, was assessing mechanisms facilitating the success of the invasive alga Sargassum horneri. Okay, so a little bit of background, what exactly are invasive species? So while many species may travel from their native range to a non-native location, species that have no real discernible impact on the environments they invade are considered to be introduced species. On the other hand, species that have a negative impact on the environments they invade are considered to be invasive species. Sorry, I need to close it. There's like a screen in here that's messing me up. Okay. Successful species invasions can often lead to the loss of ecosystem services, economic loss, reduced ecosystem complexity, habitat loss, and declines in biodiversity. So kind of transitioning onto my study species, Sargassum horneri, it's an invasive brown alga in Southern California and Baja California. As I mentioned, it's a brown alga in the class Phaeophyceae. It exhibits a rapid growth rate and a high fecundity, and it can grow up to 3 meters in length, so pretty tall. It possesses air bladders or nematocysts. It exhibits a wide temperature tolerance, and as everybody has mentioned so far, it exhibits an annual life history, so it completes an entire life cycle within 1 year. Sargassum horneri is native to South Korea and Japan, and it arrived in Long Beach Harbor in 2003. Since its arrival, you can see it's kind of spread. The left panels represent Southern California and the right panels represent Baja. And the dots represent Sargassum horneri invaded sites. And you can see how it's spread since its arrival in 2003 in Long Beach Harbor, throughout the Channel Islands, north towards Point Conception, and then also south into Baja, California. So despite its seemingly rapid proliferation since its arrival, and current widespread range, little is really known about why it's so successful in this region, as well as whether certain communities are more or less susceptible to invasion. And this information is particularly critical because recent attempts at eradicating Sargassum horneri in already established sites have proven unsuccessful. So kind of knowing what communities are more vulnerable or resistant to invasion and identifying those mechanisms that facilitate that is really important in managing this invasion moving forward. So that's where my research comes in, which is what mechanisms facilitate the success of Sargassum horneri, as well as what mechanisms facilitate community susceptibility or resistance to invasion. So there actually has been one study in this regard, and it was done by researchers at UC Santa Barbara and published in 2017. And these researchers used long-term data sets, specifically PSCO, if anybody's familiar, as well as National Park Service data from the Northern Channel Islands. And these programs collect the same community metrics year after year at the same sites. And so it's kind of a really rich data set in order to draw conclusions from. And these researchers looked at this long-term data to see what communities may be more or less susceptible to invasion. And what they found was that the most resistant communities were those that had a high native algal diversity and abundance. So they predicted that competition with native algae for space, light, and potentially nutrients was inhibiting Sargassum horneri from establishing at these sites. They also found that sites dominated by sea urchins were also resistant to invasion by Sargassum horneri because of intense herbivory. So these sea urchins were eating all the algae in sight, including Sargassum horneri, and so it wasn't able to establish there. They also found that the most vulnerable communities were those that either had an intermediate algal diversity and abundance or varying urchin densities, so neither competition or herbivory could really inhibit Sargassum horneri at these sites. So as I mentioned, this previous study was only derived from long-term data, and so there actually haven't been any experimental tests of these conclusions in the field. Also, these conclusions were only drawn from Anacapa Island in the Northern Channel Islands only, so my research specifically seeks to address or to test these conclusions in the field and also look at whether competition or herbivory can determine Sargassum horneri success on Catalina Island and the Southern Channel Islands because they're markedly different than the Northern Channel Islands. So to do this, I chose one study site, which is denoted in the yellow teardrop here. The red star represents the Wrigley Institute, and this site is Isthmus Point. And from my previous research last summer, I knew that within this Isthmus Point site, there were discrete patches of, areas that had dense urchins and low native algal abundance, and also patches that had high native algal abundance and low urchins. So it was a perfect site to test these conclusions without any, other confounding environmental factors. So within this site, I did some, mini site categorizations where I quantified native algal abundance with quadrat surveys, and then also urchin density and size with some swath surveys. And from these categorizations, I chose 4 sites, 2 of which were my algal-dominated sites, denoted in yellow, and 2 of which were my urchin-dominated sites, denoted in purple. And this is my algal percent cover or abundance data. And you can see that my 2 algal-dominated sites are dominated by algae and have significantly greater algal percent cover than my 2 urchin sites, which was great. And these algal-dominated sites were dominated by this low-lying brown algae in the order Dictyotales, if anybody wants to know. And then also for my urchin surveys, I found that my 2 urchin sites had significantly greater urchin abundance than my 2 algal sites, which was also good for testing these conclusions or these hypotheses. So once I had these site categorizations underway, I ran some growth experiments where I collected a bunch of recruit Sargassum horneri. And recruit Sargassum horneri is the smallest of the, macro life stages of the Sargassum horneri life cycle and the most vulnerable of these life stages. So if recruit Sargassum horneri can survive in these sites, then it's likely that the life cycle will proceed and the invasion will continue. So I collected a bunch of recruit Sargassum horneri, spun everything in a low-velocity centrifuge, also known as your household salad spinner. And this allowed me to standardize the amount of water on each thallus and get an accurate and reproducible wet weight. So I weighed everything, attached everything to lines, and then for half of the replicates at each site, I placed them in cages. And these cages are specifically designed to prevent urchin herbivory, but allow other types of herbivory to occur. So as the urchins kind of crawl up the sides of the cages and reach these lips, they fall off, but the top of these cages is completely open so other herbivores can still have access to the algae inside. And then the bottoms are completely open, save for a small strip of cage and down the middle, to which I attached Sargassum horneri to. And this allowed other types of algae to kind of come through the bottom and still interact with Sargassum horneri, while still preventing urchin herbivory. So placed half of them in cages, and then I also monitored light and temperature at each of these sites. But basically I put all of this out into the field for approximately 2 weeks, and then pulled everything out and then measured how much everything grew at each of these sites. And here are some pictures of my experimental units. On the left are my urchin exclusion cages, and then on the right are my uncaged replicates. And what I found, the y-axis represents percent change in weight per day or growth per day. And what I found was that for my algal-dominated sites, almost all of my treatments had biomass loss that was significantly less than 0, save for that one bar in the first algae site. And then for my urchin-dominated sites, almost everything had biomass loss that was not significantly less than 0, save for one of my treatments in my first urchin site. So running a 2-factor ANOVA on these data, site and caging treatment did not have significant effects on Sargassum horneri growth. So the fact that site didn't have a significant effect makes sense because technically all of these sites were supposed to be resistant to invasion. So there should be kind of equal, I guess, like resistance to invasion between the sites. So that makes sense. The fact that caging treatment did not have a significant effect, I'll go into in a little bit. But looking at these data a little bit further, as I mentioned, it seems like there's greater biomass loss in the algal-dominated sites just because almost everything had biomass loss significantly less than 0, save for one of the treatments. And this is not the case in the urchin sites. So it seems like maybe the algal-dominated sites are more resistant to invasion, but it's still not, I guess, like significantly different than the urchin-dominated sites, but it would be something interesting to explore further. So going into the non-significant differences between caging treatment, for the algal-dominated sites, it makes sense that there wasn't a significant effect of caging treatment because the native algae was affecting both the caged and the uncaged treatment supposedly equally. But in the urchin sites, you would expect that the cages would protect Sargassum from herbivory and that the caged treatments would have higher growth. But that's not the case. So that kind of led me to think that because there's no algae in these sites to like really affect Sargassum horneri, it led me to think that maybe there was other herbivores that were maybe eating Sargassum horneri. And upon closer inspection, I found that frequently there were these herbivorous snails in my urchin exclusion cages and oftentimes attached to Sargassum horneri. And then running surveys on these snail densities, you can see that there's significantly greater snail densities at these urchin sites than there were in the algal-dominated sites, but still pretty high in the algal-dominated sites. So potentially these herbivorous snails could be eating Sargassum horneri in both of these sites, but this is something that would need to be explored further. Okay, so I guess conclusions and implications of this study. I found that sites with strong competition and herbivory are resistant to Sargassum horneri, so this aligns with those conclusions from the long-term data. And then as far as management goes, potentially we should focus management efforts on maintaining these resistant communities in order to inhibit Sargassum horneri from spreading. But you don't really want to manage an urchin barren or maintain an urchin barren because it's an unhealthy ecosystem state. So I guess my suggestion would be to try and manage these healthy native algal communities, which I guess are derived from a whole bunch of other trophic interactions. So kind of maintaining, I guess, a healthy native ecosystem in order to inhibit Sargassum horneri. And then identifying vulnerable communities. So I guess testing those conclusions from that previous research in the field as far as what communities are more or less vulnerable to invasion. So like maybe what urchin densities, what threshold maybe would allow Sargassum horneri to really take off, and what native algal densities may allow Sargassum horneri to really take off. So that threshold between vulnerable and resistant would be interesting to explore. And then finally, further understanding these invasion mechanisms will enhance the predictability of the Sargassum horneri spread, which is particularly critical because widespread eradication is likely not feasible at this point. So kind of pinpointing where it may go next is really important. And with that, I'd like to acknowledge my PhD advisor, Peggy Fong, Lauren for the dive support, and for the dive support, and then a variety of funding sources, specifically the Wrigley Institute, which has been very critical. And with that, I'll take any questions.
Hi, my name is Louise, and I'm a second year in the Museum Lab at USC. And we've heard so far how bad Sargassum is, and today I'm here to talk to you, along with my lab mate Jose, about how great macrocystis is. It's also known as giant kelp. It forms these giant kelp forests that you see here, which are essential for a lot of vibrant ecosystems in the ocean and home to a lot of marine organisms. Here we're kind of using a different approach. I know domestication is kind of a weird word to use with macroalgae, but it's kind of becoming more and more popular as agriculture is getting decimated by climate change. We're kind of thinking of new ways to deal with providing food for us and providing fuel for us. And I think the ocean, to a lot of people, represents kind of this more untapped, you don't have to put in as many nutrients, you don't have to use as much land space. And so aquaculture is actually kind of taking off in the United States. These are just a few companies that have opened very recently. And they're kind of using, like, some of this is like the multi-trophic aquaculture that Abby and I were talking about earlier, that's looking at bioremediation efforts that also grows food and essentials for us. And a lot of these are focused more on edible kelp farming. So this is wakame, which goes in like seaweed salad, and they also, I think Blue Evolution in Alaska is infusing pasta with kelp. So find that in your local Whole Foods. And our lab has actually our own aquacultural test farm in Santa Barbara Harbor, where we're growing macrocystis on long lines, kind of hanging from anchors at the surface. And you're probably wondering at this point why we are interested in giant kelp. Spoiler alert, it does not taste good. So it's actually got great applications as a biofuel. And the limiting reagent here for using it as a biofuel is biomass. So we are hoping to use domestication to help the industry tackle this issue, because if you're sourcing giant kelp from native populations, obviously you're messing with those ecosystems and you're probably not even getting enough material from that. So we're trying to improve the crop into something that can be used and actually as a viable disruptive biofuel. So crop domestication by humans typically takes smaller fruits and plants, and we select for the things that we want to make bigger or grow taller, and we select against things that we don't want, like lots of leaves, bushiness. So this is just an example of corn compared to its ancestor teosinte, but this is obviously repeated in like the apple or orange or fig. I mean, you've seen the effects of domestication, especially if you look up the ancestral plant from which humans domesticated from. And so we're kind of looking at not necessarily domesticating giant kelp in the same amount of time as corn, because it took millennia. So our lab is hoping to do it very quickly, using modern next-generation sequencing techniques to get more information about the genes that are important in this domestication process. So a big concern so far in us trying to permit and do this sort of experimental work and in the industry as a whole is that people are very concerned about the effects of non-native or domesticated kelp species on native ecosystems. And it's actually a very valid concern. You've heard about Sargassum as an invasive species. There's also the kelp that is used to make seaweed salad, Undaria pinnatifida. They tried to grow it along the European coastline. And it is now a hugely invasive species all around the UK, France, and Spain, and they actually pay people to collect it from the beach because they hate it so much. So when we went to apply for grants and kind of get people to approve of us planting giant kelp in the world, we turned to the giant kelp life cycle to see if we could find a way to sterilize the organism. So one of my lab mates, Gary Milano, he's actually probably driving back from our kelp farm in Santa Barbara right now. He had the bright idea of looking at the life cycle, which I'm sure you got from Anne a little bit earlier, but there's this spot where it undergoes meiosis and makes spores once it's planted out in like an industry aquaculture setup. So if we can exploit where the organism is undergoing meiosis and stop that, you can actually have a plant that just starts from zoospore and goes to sporophyte and stops there and does not release any domesticated or like selected spores into the ocean. And so you actually end up linearizing the life cycle. And the way that you're able to do this is because giant kelp has this nifty quality that when you keep gametophytes under red light, they just explode. They divide endlessly, rapidly, clonally, so you just end up with a lot of seeds. Kind of like a seed stock for like apple or orange in agriculture is the comparison that we make a lot. And so the advantage of this is that you can have gametophytes that are sterilized that don't have to go, you don't have to go collect these spores to make another round of growth. You can just keep growing from this same stalk into sporophyte, and so you can have it reliably sterilized, and you can select in this pool for the traits that you're looking for in domestication. So the way that we're looking into sterilizing is through transcriptomics. So that's where some of the next gen sequencing comes in. This is a little bit of what I'm looking to do. I'm not going to go into too much detail, but basically we want to use long read sequencing on a lot of different, on samples from a lot of different tissue types, so that we can do a comparison to understand like what genes are present, like all isoforms that are present in different tissue types, and figure out which genes are involved in reproduction and which are not. So a lot of my work this summer was on the thing that you have to do before you can sequence, which is extract nucleic acids. Unfortunately, from giant kelp, it's not a model organism, and it's very thick and terrible. But we went out and collected fresh samples every day. These are my awesome RE students, Ben and Jean. And Ben got his boat certification. We would go to the same like float every day before we would do an extraction and work with fresh tissue, which was really awesome because you could like trust the material and know that you weren't working with like degraded RNA, and you could see like, okay, these things that I'm doing are actually extracting RNA, and you can trust your samples kind of more, was the awesome thing about being out on Wrigley. And some of our results were just that from the development of the blade, you get a higher concentration of RNA when you extract from segment 1. And we also found that the yield of RNA after running it through an extraction can be super dependent on which blade you choose, which was kind of weird to see, but we compared like 4 samples from one and 4 samples from another, and basically one blade just gave absolutely no RNA. So it could be something to do with the age of the blade and if it's senescing, and so we're kind of trying to delve into that more. So just for anybody who's not super familiar, the critical steps of RNA extraction are to homogenize in lice, and a big issue here is the homogenization. This tissue is like working with the toughest plant you can imagine, and so we've been beating it and freezing it and doing everything we can to break this thing, and we're kind of getting there. We're working a little bit more with our collaborator on campus here in the marine biology department, and we're kind of getting somewhere with this, which is really awesome. Another thing is that proteins that degrade RNA, which live all over your hands. So when you're working in the lab, you really have to wear gloves, spray everything, so that you don't contaminate what you're working with. And so that was like a big learning curve for my undergraduates, but I think they got it near the end. Basically these two steps, we've been trying to really work on the homogenization and adding more and more things to make sure that the RNA is not getting degraded throughout. So throughout the summer, we've had pretty consistently low RNA concentrations. We want these to be like a little bit more in like near 100 for like 100 nanograms per microliter for each sample. But you can see that so for individual 9, we're getting like very consistent, at least content of RNA, and for individual 13, we're really not seeing that much. And so something else that we're looking at is the ratios of contaminants that we're seeing with spectrophotometry. And the 260, 280 is pretty good, but we see like a huge contamination peak at 230. So we're thinking if we clean up the RNA a lot, we might be getting more than we're actually seeing from these sort of quantitative analyses, because we're using a lot of tissue and we're pretty sure the lysis protocol is working at this point. So this is just kind of a summary of unsuccessful things that we tried and more successful things that we tried. So basically anything that heats it, breaks it up, etc. And this is all pretty methods heavy, but is mostly what I was doing on the island with my undergraduates. And we got some pretty good results, and they're awesome, and they were great. So kind of the next steps that I'm thinking about is which types of tissues I'm using in this analysis, and being able to extract RNA from all the types of tissues that I'm interested in. I want to use gametophytes and non-reproductive blades, and I also want to use the sporophyll blades themselves before they release the spores, because I'm thinking we might see a different panel of genes throughout that would pop out in a differential expression analysis like this. And we're expecting to see these genes involved in reproduction that we could mutate to sterilize this plant in this type of analysis. And with that, I want to thank Sergey and Jose for being out on the island with me forever, and Carla and John for letting us use their lab space, Diane for all of her help on the island, Ben and Jean, and Andy Gracie in the Marine Bio Department for giving me his homogenizer. Thank you. What's up?
With the blades you were picking, do you know or remember if you were picking mainly scimitar blades or just blades and you were along the front?
It was more that we were just going for material for a while. I think from now on we're gonna because we started seeing such a discrepancy between individuals, like blades, I wanted to kind of pull out more of it and see where we're sampling from along the frontispiece, if that makes a difference.
Yeah.
Yeah, and I'm assuming the ones that are farther down are going to be, more of them are senescing and getting eaten, so those are not the best.
The ones closest to the whole fast are senescing, older,
Right.
whereas the scimitar blade is the growing tip, so they can,
Mm-hmm.
There's paper.
Any more questions?
Anybody else? Any other questions?
Thank you.
Good job.
Swear, I think it's the last Cal talk of the day. It's not the last algae talk today. So my name is Jose Diesel, and I'm also the Sergey's lab. And since you heard from Kelly, basically what we're trying to do is we're trying to plant giant kelp in the open ocean, farm it a lot so we can make biofuel out of that. This way we can sink down a lot of that carbon in the atmosphere instead of using fossil fuels, right? So what I did this summer at Wrigley was just a very small step towards this, but it's for sure a very interesting one. So I tried different plant hormones on giant kelp, and I know that at the beginning it sounds a little bit weird, like plant hormones and kelp, but I hope that by the end of this presentation you're going to understand why and how we're going to use it and why kelp is important. So basically hormones are very tiny molecules that they control everything in plants. They control how they grow, they control when they don't grow. And they're basically on the top of this cascade of reactions, right? So they control all those things. And we use hormones in agriculture very, very often because if we can control how a plant's going to grow, we can basically use that in our advantage to make, bigger grapes or to make plants that have more ramifications, so we can use that in our advantage. And I know you're wondering, okay, so what about kelp? What about seaweed? Well, people have been using kelp, and so this is a picture of like kelp flower or something that people use as a fertilizer. And okay, and it works. It works really, really well. And the main idea is that it's not that you're adding nutrients with the kelp. You're adding a little bit, but you don't need that much kelp to make a plant grow. So there must be something else in that kelp that makes the plant grow. So people started looking at, well, hormones. Maybe kelp has some similar hormones to plants. And it sounds kind of crazy because we're closely related to fungi. We're more close, sorry, my English sometimes doesn't work. We are closely related to fungi? No. Well, we're closer to fungi than kelp is to plant. But people took a look and they made some genomic surveys, especially in red seaweeds, and they found some sequences in red seaweeds that were similar to plant sequences that were responsible for producing those hormones. And they also did some mass spectrometry, and they found out that, well, there are some molecules that look very, very similar to those. So can we take all of this information and use those hormones and use this technique that they use in agriculture the same way in kelp, to plant kelp in open ocean? And, well, I guess you guys know why we're dealing with kelp. So I wanted to show you a little bit of a kelp farm. And it sounds kind of weird, like how are we going to deploy hormones here? But to get to this stage where kelp is long enough and is going to grow a lot, it has to go through hatchery, which is a period where basically you incubate kelp in the lab so it grows a little bit. Then you put the kelp in those lines, and then you outplant the lines in the ocean, right? And that period is very crucial. We want kelp to be healthy when it goes to the ocean. We want it to be big enough because the bigger the kelp is, the faster it's going to grow. And at hatchery time, kelp is so tiny, it's not going to grow a lot. So if you can accelerate that period, it's going to be awesome. So basically, the main goals that we had is to reduce this hatchery time, and maybe we can find some hormone that's going to make some morphological manipulation that we might be interested in doing before outplanting. So this is basically a kelp hatchery. That's where you grow them a little bit before planting. Does that make sense? Perfect. So this is my experimental setup in Catalina. And I like it kelp. I call it kelp spa because it's literally a bubble bath with controlled temperature. Basically, what we all need on a Friday night. So I tested 4 different hormones, and they are in this big tub in a temperature-controlled environment. So I have fresh water under the buckets, and that's between 15 and 17 degrees Celsius. And I put kelps inside those buckets. And basically I tried 4 different hormones because I was going for everything, right? We don't have a lot of information on this, so this is basically a trial to see what works. And they stayed there for between 4 and 6 weeks. So they stayed there for 24 days. Every 4 days, I would take the water out of the buckets, measure the kelp, put the water back, and introduce the hormones again. So our expectation is we're going to see kelp growing, we're going to see it changing, and we can also do some computational experiments after kelp is already grown. And well, we got some interesting results. So every time I changed water, I counted as a time point. We had 7 time points at the end of this. And on the 5th time point, I decided to add some more media, right? And it was really interesting. So basically, by the end of the experiment, we noticed that an ANOVA test told us that the hormones that we were using were significant. So we had changes between the hormones, blade length is also significant, and this shows how important the hatchery time is. This showed that the bigger the blade, faster kelp is going to grow. So during that hatchery time where kelp is very, very tiny, it's not going to grow a lot. So it's very important that we can just make that thing grow fast. Also, concentration seems to be important. I tried 3 different concentrations of hormones, usually based on how they're used in agriculture, going for the minimum concentration that I saw on papers and the maximum one and something in the middle. And I also found no difference between blades. That's expected. That kind of tells you that the experiment worked. And a 3D test shows that during this first 5 time points, the only hormone that showed difference against control was IBA. And it was a horrible difference because all the plants just died. They just disintegrated. So it didn't sound very exciting at the beginning, but it gets better. So I added media, it's called F/2, and it was not a lot, but somehow we guess that it changed the microbiome community. Basically, every kelp plant started dying. We're like, whoa, what's happening? But every plant started dying, but in one hormone, and that was abscisic acid. Basically, abscisic acid showed positive growth throughout the whole experiment, right? And this was very exciting. It's basically the result that we're trying to see. So abscisic acid in plants is responsible for controlled stress. So basically, they make plants stop growing so they can manage stressful situations. And adding the media was a stressful situation, right? This is just an example of how things grew a little bit. So the IBA, everything died. The abscisic acid, everything grew. So it didn't grow faster than the controls, but it kept growing regardless of the addition of media, which was a stressful situation. And with that, I want to thank everyone that participated on this, especially our U students, and Kelly, and the lab. And any questions?
Is there, do the hormones ever, is there a lifespan of the hormones, in the water?
See, we were changing water every 4 days, so yeah, they're definitely going to use the hormones, but those hormones are used in such a low concentration that it's just a tiny bit. But yeah, you expect that to happen. They're going to be used up.
So if they're I don't know if Elizabeth's here, but because we were talking to Ben during this poster, and she was wondering if these hormones are being used in terrestrial agriculture, is there a possibility they could run off and affect that kelp communities, nearshore kelp communities or something? Do the hormones kind of denature at some point, or I
think they definitely get used up, not only by kelp but by other microbes that are there. We didn't see a lot of, so we saw differences in concentration of the hormone between experiments in the first 5 time points, and that shows that, how can I say that? That yes, increasing the hormones are going to have different effects. So we didn't get to a point, unless abscisic acid, we definitely got to a point where it's saturated. We had no difference between concentrations. So a tiny bit of that is just enough. And if you add more of that, it's just not going to make a difference. And it might as well just sit in the water. So, but it's something to see.
Did they change color after you added media?
They basically got extremely slimy, and you just pick them up and they just fall apart. They were just degrading. Yeah. But the one with abscisic acid. Any more questions? Thank you.
All right. Hi everyone, my name is Catherine Scafidi. I'm going into my 3rd year of my master's program now, so this summer kind of finished up my field work, and now I'm going through some samples in lab and data processing, but I'm at California State University Northridge under the advisement of Dr. Mark Steele in the fish ecology lab. So I'm going to be talking about fish, but then there's also this important invasive alga that I'll gloss over because you guys are all professionals now. But overall, I am looking at the effects that an invasive alga has on the trophic dynamics of our temperate rocky reefs. Is it? Oh, okay, that helps. All right, so overview of what trophic interactions are. I'll use some land-based examples and then I'll kind of go into the marine example. But we always usually start off with a primary producer, such as this grass, and then we have some sort of primary consumer that is directly feeding on the primary producer. Then we also have secondary consumers, like this little mouse that'll eat the grasshopper. And then we get all the way up to the tertiary consumers, which are typically our big apex predators like eagles, wolves, etc. And so in the marine environment, I'm focusing mainly on this interaction of the primary producer, primary consumer, and secondary consumer. And so for my study, my primary producer is going to be algae, specifically the invasive alga. And then the primary consumer are these small little invertebrate epifauna that live on and among the algae. They're small little crustaceans, snails, things like that. And then my secondary consumer is going to be, or my main predator in my system is going to be one of our local reef fishes, which is the rock grass. And so what I'm interested in, like I said, is this interaction that we get between all of these three levels, but also what the indirect effects are and interactions of the fish and the algae. So I'll go through kind of this chain reaction, but again, the overarching idea is what is that second level interaction beyond the initial ones that people have been exploring. So this map shows, I like this map because it's a great visual to show you that all the white lines around here are the different shipping pathways that our big freighter ships use around the world. So from this map, you can see that almost no coastline is untouched at this point. So we have this really vast amount of interconnectivity across our oceans and even sometimes really far into our waterways, reaching more land restricted areas. But what we can see here is, for my research, is over here is the coast of Japan, and then we are down here off of Southern California. And so there's this huge open ocean that organisms need to cross in order to get across to invade. And what we have in our local port of LA is it's one of the biggest ports of our country. So it sees thousands and thousands of shipping freighter ships coming into the port in a year. And so that has increased the susceptibility of our reefs and our local waters of getting these invasive species, such as, you guessed it. I'll just throw these up here really quick. Native to South Korea, Japan, and China. It's annual brown alga. It's monoecious, so it can fertilize. It's capable of self-fertilization. And the big thing too down here is they contain these secondary metabolites called phenolic compounds, and this is actually a trait that all brown algae have, which I'll get to later on. And you already saw this map through a few people's presentations, but I went into this animated route here and got really excited about it until it became really tedious. But you'll notice that since its invasion on Catalina Island in 2006 is when we first discovered it on the leeward side. And so what's going to happen is as the years go by, you'll start to see how it's spread along our Southern California bight. And unfortunately, this has become a very strong invasive in the southern waters and then also down into Baja California, but I don't have a map of that right now. But I really want to point out how quickly it has expanded its range along Catalina Island itself on that leeward side, and up to 2015, even onto the west end side, we're seeing some of it. So it's really good at spreading, is the moral of that slide. So my question was, what effects does Sargassum horneri have on these multiple trophic levels in the rocky reefs? And so my expected effects were that I was going to use these three metrics to test this. And so the first one is I used fish foraging observations to see if the fish were even associating with the alga. From there, I would quantify the invertebrate epifaunal abundance on the individuals of the invasive compared to some of the native algae. And then I am working on extracting the total phenolic compounds from the invasive and comparing that to the natives. Now, I expected that since it was invasive, it was new, the fish weren't finding food and they weren't foraging among it as, if at all, but as frequently as with the native algae. And that would be because there's a lower abundance of their food source. And that is due to the higher concentrations of those phenolic compounds, so it's making it unpalatable for those primary consumers. So my study sites were, oh god, we're not at SCAS, sorry. We're at USC, same spot. My study sites were off the coast of Southern California on Santa Catalina Island, and specifically on the leeward side. So right here we have the Wrigley Marine Science Center, Two Harbors is over here. I had 6 sites ranging from Arrow Point down here, up here, down to Empire Landing to kind of get a broad distribution of these sites and to see if anything that I find would be, wouldn't be site specific. So the first part of my field work, I did this last year, and I'll get into what I did this year, but I decided to observe 3 of our common reef fishes. So we have rockgrass, California sheephead, and Garibaldi, because in my preliminary observations, all 3 of these species forage very similarly. They're picking off the algae and they're in the same depth range, and they have pretty high site fidelity, so they don't tend to cross from reefs over large sandy bottoms to other reefs. So I can ensure that the sites that I chose, they're not showing up at Arrow Point and then traveling all the way down to Empire Landing in the day. I did benthic surveys of my sites where I ran out transects at sorry, it's been a while, at between 10 to 30 feet, because that was where I primarily saw all of those fish species cohabitating. So I ran out these transects and did point contact method just to quantify the percent algae that was there. So I could say these fish were foraging among this, and this is what was available to them at these sites. I then did some algal collections. This was done last November. So I had the invasive Sargassum horneri, and I'm comparing them to the native Sargassum palmeri, Dictyopteris undulata, and Zenaria farlowii. These 2 are in the same class, and then these 2, or order, and then these 2 are in the same order as well. So that'll kind of come into play with my chemical stuff later. So I collected individuals of the algae. I collected 15 individuals per site, per species, and they were collected in these little fine mesh tea bags, so I could ensure that the invertebrate epifauna that were on that individual at that time were collected as well. I took them back to the lab, and I rinsed off the inverts, where I then started and am still looking at them under the microscope and identifying them and counting them up. And then I took the clean algal individuals and I dried them out, and then I used a method known as the Folin method, where I extracted the phenolic compounds. And so again, I've done some of this. I'm still working on my bigger data set, because there's a lot. And this was, again, these are the total phenol extractions, so there's kind of different chemicals coming into play with that. So just kind of a rough, this is an NNDS plot, which I used on my habitat data, so you can kind of see the algal characterization at my sites. And so
when I first ran it, came out here. So what you see grouped together are more similar, and when they're grouped far away, then those are more dissimilar. So my sites are all by colors, and then these are their standard deviations from their points. And so I was like, all right, this is cool. There's some separation here. What's going on there? It's probably going to be Sargassum horneri. Plot twist, it wasn't. It's kind of out here, and it's a little bit different from here, but these are those 4 algae that I'm sampling, because these 3 are the most common of the understory brown algae there. And so it's interesting to me on this to see that they're more similar, but what was driving the site differences was the presence of kelp. So I had only at 2 of these sites where they're actual kelp forests. So that made sense to me because then I saw a more diverse algal presentation there. So for the next couple of, so those are my top 4. The next 3 graphs are going to look very similar. On the x-axis, I'll have algal species, so I have the 3 natives and then my invasive over here, and then the y-axis will change. So for this first one, this is the foraging rate. This is the number of bites per minute, and it's all the fish species pooled. But you can see just overall that they really weren't foraging among the invasive compared to the native. I'm pretty sure that only Garibaldi were the ones taking bites off of it, and they're kind of weird, so I discount them. No, they're important. But the big thing too is that you see its native congener has this high foraging rate, which could be due to its structure. We'll kind of get into later. But so this was kind of cool. I ran this and I was like, all right, they're not feeding among it, so let's see if their food is there. So again, algal species on the x-axis, and then I have epifaunal abundance, just overall abundance on the y-axis. And okay, that makes sense. They're not really there, so that's why. I saw this visually just when I was rinsing it off. There was barely any inverts coming off of it, but then the stats to back it up are really great. So Sargassum horneri had the most, and that could be due to the alga itself is very branching, and there's a lot of spacing in there, and it also has a lot of epiphytic algae that grows on it, so it creates a better habitat for the small little inverts to live in. And then these 2, they have they have some similar inverts that are living among them, but they tended to have more clams and snails because their blades are broader, so there's more surface area. But there's still close clusterings of the blades to create a good dense habitat for them to hide in. And horneri is, it's kind of plainer, the fronds itself, and it looks as branched as palmeri, but they just still weren't hanging out even though I got adults that were really, really bushy compared to, say, like a recruit that may not be as complex. So this is where my phenol extractions come into play, and I'm thinking that maybe there's something chemically going on that is making those inverts just not want to hang out there. And so again, my algal species on the x-axis, and this is my absorption values. So the greater the value, the greater the concentration of total phenols were in the algae. And so this one wasn't statistically significant, but again, I only had a sample size of 7, so it's still small. I have like 300 left to go through. So, but you can see already that Sargassum horneri has a very low absorption value compared to horneri, Sargassum horneri, the invasive. So that to me was like, okay, that's it. Eureka, that's the reason why. But these 2 other natives have very similar, if not more, concentrations of their total phenols. And so I'm still kind of delving into why this is. And from some papers that I've been reading, I need to kind of figure out, for my research, I won't be isolating each phenolic compound because that's a lot. But it could be due to, again, these are in a different order compared to the Sargassum species.
And so they're in the order Dictyotales, and it could be that there is another phenol that is driving those increases, but it may not be one that is attributed to deferring the inverts from it. So something that they're foraging on, this could be a phenol could be for sun protection or something like that that's driving that trend. So I'm still looking into that, but still interesting to see that the two Sargassum species are so different. So overall, what have I found so far? I found that the fish foraging was lower among the invasive, and that was due to the lower abundance of inverts. And I found an increase in the phenols compared to the native, but not to the native Sargassum, but not to the other two native species. And again, I'm still kind of exploring that. And what I've been doing this past summer, I was doing my benthic surveys as well, and then I've been catching fish. I collected 300 of them last year, and then I got another this year, where I've been collecting the females to look at their, I dissect their otoliths, their inner ear bones out, and their gonads, so I can assess their growth rate and their reproductive output to see if that's being affected by their sites. So if their food source is limited, is that affecting their growth and reproduction down the line? So that's all to come in this fall. And overall, why do we care about this? It's good to know that if this species, this invasive, doesn't provide a comparable habitat for the fish prey, that we have to adjust some future mitigation efforts. And just in general, knowing how an invasive alga can kind of shift these trophic interactions is important for mitigation effects. And it's also important to look at these invasion events at a whole trophic interaction level, so you can kind of be able to recognize certain sites that are susceptible, like what everyone before me, Emily and Lauren and Anne, have been talking about, is, you know, more complex habitat is more resilient to these invasions, so we can kind of assess what could happen in the future. And again, this is just understanding invasive species and how they are cohabitating in these invaded areas can help us figure out ways to put in policy that can change, you know, the shipping pathways or different measures that are out there to try to prevent future invasions, because once it happens, it's kind of hard to get rid of them. So with that, I'll say thank you to everyone who has helped me. It has taken an army, lab mates, fellows, and some undergrads, and my research facilities. Wrigley has been a huge part in this research, and I really appreciate everything and this fellowship has offered me, and my funding sources. Take any questions.
Okay, so today I'm going to be talking to you guys about my summer research project, which was looking at thermal tolerance of the symbiotic anemone Anthopleura elegantissima across environmental gradients. No? Okay. So Anthopleura elegantissima is a sea anemone that lives in the intertidal zone. It's a pretty cool system to work on because it reproduces both asexually and sexually. So if you look at the figure in the top left there, that's an aggregation of over 100 individuals, and all of them are clonal. However, they do reproduce sexually as well through broadcast spawning, so discrete aggregations are genetically distinct, and there is quite a bit of genetic variation that you can find within a single site. They're also pretty interesting because they engage in symbiosis with the dinoflagellate Breviolum muscatinii, where the host essentially provides shelter for the symbiont in return for photosynthetic products that give it energy. However, under stressful conditions, this partnership breaks down. So it can result in a loss of the symbiont cells themselves or the photosynthetic pigments of the symbiont in a process known as bleaching. So this is really well known in tropical corals. and can occur only 1 or 2 degrees Celsius above the average temperature. In contrast, Anthoplura elegantissima experiences temperature fluctuations of up to 20 degrees Celsius during tidal exposure. So, I'm interested in the genetic and environmental factors that may lead to increased thermal tolerance. And to do this, I'm going to be making 3 main comparisons at very different spatial scales. So the first is across a latitudinal gradient, so a site comparison. So I have a site that's in the north in Malibu and a site on Catalina Island, which would be my southern site. And so my hypothesis would be that the site on Catalina is going to be more thermally tolerant than the more northern site. And this will most likely be due to just fixed genetic differences based on population structure. The second is within each site across the intertidal zone. So the intertidal forms a natural vertical gradient, where as you go higher in the intertidal zone, you have increased temperature fluctuations versus lower in the intertidal, where the anemones will be mostly submerged at a more constant, lower temperature. However, if we do see this pattern, it could be due to either fixed genetic effects or plastic effects due to acclimation. So there may be selection across this gradient for more thermally tolerant alleles, but it could also just be that anemones sampled from the higher intertidal are acclimated to a higher temperature and thus shift their thermal performance. The last comparison is within a single aggregation. So within an aggregation that's all clones, a previous study found that anemones on the outside of these aggregations actually have higher body temperatures than ones on the inside during tidal exposure. So we'll be comparing thermal tolerance within these aggregations to ask questions about the plastic effects on thermal tolerance just due to the thermal history. So, although we have an idea about the thermal gradients these anemones span, no one has actually experimentally induced bleaching in Southern California populations. So the first thing I had to do was sample some anemones and expose them to 5 different temperatures. So here on the x-axis we have temperature, and then chlorophyll on the y-axis. And not surprisingly, you can see as you increase temperature, chlorophyll is decreasing. So these anemones are bleaching. And you can see this visually up here. So the healthy anemone up top, and then the one on the bottom has lost its symbionts and it is bleached. So although this is the response I was looking for, I needed to choose a temperature where we're gonna see this bleaching, but also maintain variation within the population. So I further split up this graph into anemones sampled from the high intertidal in the pink and the low intertidal in the blue. Does not look like those colors, but they are. And so again, we have temperature on the X, chlorophyll on the Y. And this is actually exactly the opposite of what I expected. So it seems here that the anemones sampled from the low intertidal are actually more thermally tolerant than those sampled from the high intertidal. However, this was a pilot experiment, so it was really low sample sizes. This also doesn't really tell us anything about the mechanisms that are underlying this response. What it does tell us is what temperature that I could use for my full-scale experiment. So if you see between 27 and 29 degrees, you kind of see the low and the high separating out. So I chose a temperature within there to use for my full-scale experiment. So a couple weeks ago, I went ahead and sampled anemones from 2 sites, Point Dume in Malibu and Shark Harbor on Catalina Island. And I sampled them from both the low and high intertidal, as well as inside and outside of the aggregations. I brought them all back to Wrigley and gave them a 10-day acclimation period before I put them in their experimental treatments. So a control temperature of 20.5 degrees, which was the ambient water temperature at sampling, and then a heat treatment of 28.5 degrees, which I determined from those pilot experiments. Throughout the experiment, I was actually able to measure effective and maximum quantum yield of the photosystem of the symbiont. And so this is essentially a metric for how efficient the symbiont is photosynthesizing, as well as if there is any photo damage to the symbiont. And so I did this daily using a diving PAM. So this is just some of the preliminary data that I got. So this is the maximum quantum yield on the y-axis that I just talked about, and then day on the x-axis. So you can see that the pink line is decreasing over time, which is the heat treatment. So there is photo damage to the symbiont, which is a good sign. That means the experiment is working. And there was a significant effect of both treatment and day. However, this doesn't actually indicate bleaching necessarily. It just means that the symbiont itself is impaired. So since I only did this a couple weeks ago, I haven't had a chance to really analyze that data yet. But the next steps are to, I destructively sampled them, and now I'll look at symbiont density and chlorophyll, which will give me an idea of bleaching as well as the mechanisms that might underlie bleaching. I will also be genotyping the host to look at population structure between sites as well as within a site, and the symbiont itself. So the symbiont can be more or less thermally tolerant, which would influence bleaching as well. And so the last thing I'm looking at is gene expression. So looking at differentially expressed genes within an aggregation, again, trying to look at plastic effects versus between different genotypes. So I just want to end with kind of reiterating my expectation. So I expect that the anemones sampled from Catalina are going to be more thermally tolerant than ones from higher latitudes in Malibu. I do expect to see this countergradient variation across the intertidal that we observed in the pilot experiment, and hopefully get a little more insight into why this might be occurring, if it is genetic or it is just due to acclimation. And lastly, that anemones on the outside of aggregations will have higher thermal tolerance than those on the inside, just due to acclimation. So with that, I'd like to acknowledge my PI, Carly Kenkel, my undergrad, Connie, and my lab mate, Wyatt, for helping me with sampling, as well as Kelly and the rest of the Wrigley staff for supporting me during my experiment, and the Wrigley Backus Fellowship that funded this. Yeah.
So why would it be expected to be more thermally tolerant depending if it's within or outside of the aggregation? Is it just from albedo?
Just from so the ones on the outside, they're just being exposed to higher temperatures than the ones on the inside.
Is it just because the albedo of the colony versus the substrate, or is it something else?
No, it's because like there's I think it's because it's being like buffered on the inside, because they also hold a lot of water in them. So on the inside you have that like buffering system versus ones on the outside that are just going to heat up more.
Yeah. So same question, but for Catalina versus Malibu, do you expect Catalina to be more thermally tolerant as your hypothesis? Right?
Yes. And why? Because so that's a good question, and something that I do have to do is put out temperature loggers where I sampled these anemones from. So I actually only have temperature data from Catalina right now, but looking at just like water temperature and air temperature data, the Malibu site is not as warm as the Catalina site. So just based on.
Just in general?
Yeah.
Because I was wondering, because you had one slide where you saw the opposite trend where the deep sample ones were more thermally tolerant. But if you think about it, if they're still in the intertidal, the deeper ones are going to be experiencing the coldest water, but also still sun exposure, right? So maybe they have to have a wider range, right? So
well, they all will experience both. They're not, none of them are, they all will experience the low temperature, will be submerged. They're only really, so I have like some temperature data here, So there's like really extreme fluctuations, and this was like in the middle of the intertidal that I did this. So you can see like when there are the really extreme tides is when you're getting.
The whole population sees that.
Yeah.
All right, hi guys. My name is Elizabeth. I'm Elizabeth, that's how you pronounce my name. I'm from Cal State Northridge, and I just did my first year of grad school. Woo, survived. Today I'm going to be talking about the verification of giant sea bass spawning through auditory and visual observations. So before I do that, I just want to let you know about a little bit of my study species, and then I'll talk about my question, and then some stuff we know about giant sea bass, like our background, what we think we know. Then talk about the questions, methods, and then what results we got. So giant sea bass are critically endangered and they're the apex predator of the giant kelp forest. They're the largest bony fish off the coast of California, and you can find them from south of Point Conception all the way to Baja. They weigh over 270 kilograms, and they're long-living, living over 76 years of age. They are slow-growing, sexually maturing at ages of 11 to 13 years. Giant sea bass became critically endangered when they were overexploited due to overfishing in the early 1900s. And in order to protect them, in 1982, a moratorium was placed on giant sea bass, prohibiting the take of any giant sea bass recreationally. This was then later amended in 1994 to ban gill nets and also limit commercial catch, incidental catch, and commercial selling. In 1996, unfortunately, it was a little too late, and they were classified as critically endangered. So over here, you can see just showing how much was harvested off of them in the early 1930s. Over here is when we put in the moratorium for recreational catch, and it caused a little bit of a spike in population, but it wasn't enough until we put the gill net ban in. And as you can see, actually there's a slow, steady increase, which means not all is lost. Yay! So they have started to slowly increase in population size, and this has led to led into increased studies about them. So right now, what we know is that they actually tend to spawn. Some of their spawning aggregations tend to be happening off of the Catalina Island, and it happens between June and August at dusk between 19 and 2100 hours. But unfortunately, we still don't understand a lot of giant sea bass behaviors. And that's what makes it really difficult for us to understand what they're doing and when they're doing and why they're doing what they're doing. So there's two behaviors that we actually know about that we believe is linked to the reproduction. They produce sound and they have courtship behaviors. So what do giant sea bass sound like? Well, they actually produce a boom at a frequency of 40 to 80 hertz. And they usually do this at other male giant sea bass or any nosy divers that get too close. Before their vocalization is observed, they actually will display very aggressive tendencies. You can see him displaying his fins and making himself looking larger. He's already large, but even larger. So right before that happens, let's see if this works. You can't hear it. There's a boom. Just trust me. There's a nice little boom. So right before that happens, there's a boom, and that causes all the fish in the area to leave. We believe that because it makes other male giant sea bass leave, that we can link this to reproduction because it's showing territoriality and competition amongst other males for mate. So we believe this is done during courtship. So what do giant sea bass courtship look like? Well, from our lab, we have previous research that has hypothesized what we can see. So it starts with a female resting on the ocean floor at 10 to 30 meters. The male will then approach and she will swim deeper if she finds him to be a worthy mate. They will then start to circle each other on the seafloor, then start moving up through the water column. As this is happening, the male will nudge the female's abdomen and signaling the readiness for her to release his gametes and for her to release her gametes. They will do this and they'll spiral and they'll continue to surface where they may release their gametes if she feels that he has done the deed properly. If not, they can swim back down to the bottom, and they'll do this multiple times before they actually release any gametes in the water. But unfortunately, even though all this has been observed multiple times, we've actually never seen it result in spawning. So we've never seen the actual release of gametes in the water. We've seen this multiple times, and we've seen eggs, we've collected eggs at the top, but it's never seen the whole picture together. So why do we care about giant sea bass? Why do we want to know about their behaviors? Well, they're ecologically significant. They're at the top of the trophic level in the giant kelp forest. A giant sea bass is also economically significant because it's worth $12,600 per caught fish. That's $13 a pound. Live giant sea bass actually are very important too. They contribute $2.3 million to California's economy every year, and that's just for the chance of diving with a giant sea bass. So divers will spend $20 to $30, not even guaranteeing that they'll see one. So clearly they are important, and we still don't know a lot about them. We don't know a lot about their reproduction or what their behaviors are like. So that's what I aim to do today. So because this is just my first field season, I'm only going to really talk about these two questions, about if they produce sound during spawning and what does their courtship look like. Eventually in the future, I hope to tie these two together with a third one. So stay tuned. So these are my sites that I looked at. Is Goat Harbor, Casino Point, Little Farnsworth off the coast of California, coast of Catalina. You can see the little mini giant sea bass here indicating that. This, how we capture the sound is we take a hydrophone, and it's an underwater microphone called a sound trap. We deploy it at between June and September, and we place the sound trap at 10 to 30 meters deep on the ocean bottom, and it continuously records for 24 hours. So because giant sea bass spawn at dusk, they spawn in low light conditions, makes it hard to see the actual spawning event happen. So because this is so difficult to observe, I got a ROV. Meet Bruce, right there. He's a remote operated vehicle. He is equipped with lights, low light cameras, and a 150-foot cable. So in order to observe courtship and sound production, I'll pilot the blue Bruce from a research vessel at dusk and record and observe courtship and spawning happening at the same time with the end result of gametes being released into the water column. I will then use the hydrophone that has been placed at the site where I am recording to then verify spawning in general. This will help me understand what the reproductive event is like. So, what did we get so far? Well, we got a really big surprise. We got a lot of babies. What do I mean by we got babies? At the Southern California Marine Institute, where we have four adult giant sea bass, actually 3, because one's still juvenile. They actually spawned this year. So I got a frantic phone call over the summer saying, get your butt over here. They spawned, we have babies. And I ran over and I deployed the hydrophone and my GoPro in there. So that meant that my time was split 50-50 between Catalina and San Pedro, going back and forth to collect data. So this is what they look like. As you can see, they're pretty tiny and large at the same time. They're large eggs for a fish, but they're also very tiny for other things considered. So here's the babies. Yay, babies. This is what they look like when they get to be at 3 inches. Way cuter than the adult, I think. But don't tell Larry I said that, my advisor. This is actually a video of intake observations of what we did when we put when I put the GoPro in there. As you can see, we have circling happening here, and I have hours and hours of footage to go through. Yay! So this is showing some behaviors that we believe is linked with courtship, but we don't actually understand because there's a male and 2 females in there, and it seems that one of the females comes in and breaks it up, like, hey, that's my man, please don't spawn with him. But we're not really sure. This is the little that's Oliver over there. He's a little juvenile. He's just trying to stay out of the way of all the craziness going on in the tank. We also got, which you guys won't be able to hear, unfortunately. I wish I would have brought my speaker. I didn't know. I'm sorry. So what you're supposed to be hearing is a snare drum sound. That's what we're kind of going with it. I'm not sure if it really is like a snare drum, but anyways, you can see that you'll have a big boom here, and then it kind of peters out with a brump, brump. So you hear that, and we heard, the reason why we believe that this is a sound linked to spawning is because this sound was recorded at the time when, at 10:30 at night, and we found eggs the next morning. So I also have hundreds of hours of audio files that I will then go through with Audacity and find that out. And that's something else to show that, oh, this is what we believe is happening. So that's a sound that's never been observed or described before. And this is actually showing how loud it is. So our microphone, our hydrogen peaked out, so it couldn't even get all of the sound. It was cut off, and it was unable to capture everything. So what does this mean? Well, next year, I hope to take these things that I've observed in the tank and apply them into the wild and go out and use them to verify spawning in general. So what are the implications for this? Well, since giant sea bass are slow growing and long lived, it takes them longer periods of time to recover the population. In recent studies, it's shown that fish calls can actually use an accurate census to take how many fish there are in a population size.
By identifying these sounds that giant sea bass produce, we'll be able to use them to estimate populations and identify areas of use. This will help track population sizes and where they go, and then it will help us understand the important life history event and help us protect areas that they actually use so we can keep causing their populations to increase. So I'd like to thank my lab, the fellows at Wrigley, the Wrigley Fellowship Summer Program. I would not have a project without them, so thank you very much. Family and friends, the Wrigley Institute, the staff at Wrigley who were very, very helpful, and any dive buddies that came with me to help me out this summer. So thank you guys very much. This is a postcard I found from Catalina, circa in the 1900s. That is a giant sea bass, and that's a bluefin tuna. That just shows how prominent they were and how people wish they could ride them. I don't know, it's really cool. I want to see sold again. That'd be really awesome. Thank you guys so much.